Namaste, good evening again. Thank you for being here for this program that promises to be very, very knowledgeable, I'm sure enriching for all the people who've come here, thanks to the dignitaries and industry experts that we have with us. Thank you very much, sir, all of you to be here. So I officially welcome you on behalf of Kesar Control Systems Private Limited. It's my honor to welcome all our distinguished guests, as I said, the industry leaders, scientific experts, and esteemed delegates to today's seminar. Thank you again. Just a little bit about Kesar Control. For over 2 decades, ladies and gentlemen, Kesar Control Systems has built an enduring legacy as a trusted provider of high-precision environmental test chambers and stability storage equipment for the pharmaceutical industry. Committed to quality, excellence, global regulatory standards, and continuous innovation, Kesar empowers organizations to achieve compliance and safeguard public health. Looking forward, Kesar remains dedicated to driving advancements in automated testing equipment. Now, to move on to inaugurate today's proceedings with light and wisdom, I invite our honored guests to step forward, please, for the traditional Deep Pragati ceremony. Sir. We have with us, thank you, Mr. Sudhir Vaid, Chairman and Managing Director from Concord Biotech Limited. हम तालियां सभी लोग बजा सकते हैं लास्ट तक, अलाउड है, ये बात हुई. Thank you, Mr. Vaid. We have Dr. Viranchi Shah, promoter Saga Life Sciences Limited and former National President IDMA. Dr. Srinivas Savale, CEO AIC LMCP Foundation. We have with us Dr. SARANJIT Singh, as you all know, keynote speaker and former professor NIPER Mohali. Thank you very much. We have Mr. Mehul Patel, founder and technical director from Kesar Group, and also Mr. Jagdish Patel, Managing Director at Kesar Group. And now I invite all our dignitaries to kindly step forward to light the lamp and seek the blessings of Ganesha. Thank you everyone. Om Ganeshaya Namaha. हम आगाज करते हैं, शुरुआत करते हैं आज के कार्यक्रम का. But before we do that, I request everyone to kindly stand for our national anthem and our national song.
पंजाब सिंध गुजरात मराठा द्राविड उत्कल बंगा विंध्य हिमाचल यमुना गंगा उच्चल जलधि तरंगा तव शुभ नामे जागे तव शुभ आशीष मांगे गाहे तव जय गाथा जन गण मंगलदायक जय हे भारत भाग्य विधाता जय हे जय हे जय हे जय 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 हे
Thank you, everyone. Our first guest of honor today is Mr. Sudhir Vaid, Chairman and Managing Director of Concord Biotech Limited. Ladies and gentlemen, a distinguished biotechnologist and technocrat entrepreneur. Mr. Vaid brings nearly 50 years of biopharmaceutical expertise. After earning his microbiology degree from Punjab University in 1974, he held leadership roles in major domestic and multinational biotech firms. I request Mr. Mehul Patel, the founder and technical director of Kesar Group, to kindly felicitate Mr. Sudhir Vaid. Thank you very much. I request Shri Sudhir Vaid now to kindly share a few words with this august audience, please.
All the dignitaries on the dais, Dr. Saranjit, Dr. Viranchi Bhai, Dr. Srinivas, Mehul Bhai, and all the delegates from different industries. I think today is a very special day. I'm very happy to be here. The reason why I really want to say that it is a good time for me to be here, because I know Mehul Bhai from the day he started his business. I don't know, maybe we were the first people to have his equipments, 2007 or 2008, I don't remember. But we had a confidence in him that definitely we are going to buy a good equipment. And at that time he was working personally. I have seen him coming to the factory and doing the job personally. So I'm very happy that he has reached to this level. I was just seeing the video he sent to me, that now he is present in 65 countries. It's a good job, congratulations. And he has sold almost 7,500 equipments till date. And then he sent me one WhatsApp that 250 plus equipment are in Concord. So I was really very happy that almost 3 to 4% of the equipment what you have sold are in Concord. It is not question of selling the equipment. Basically, what he sells number of things along with that. Like, you know, the type of documentations we get from Kesar, IQ, OQ, PQ. Our all plants are basically US FDA, PMDA, Japan, EU, GMP, TGA. Means all you know, we have regulatory approvals. So it is not question of only equipment which give you a regulatory approval. Basically, these are the documents which give you, along with the equipment which give you this. So we have been very happy that Kesar has been giving us all the required documents through which we could get all our approvals. We have number of equipments, all BOD incubator, you name it, we have whatever you are making, we have that. So these approvals have come basically due to the total package which Kesar is giving, all IQ, OQ, PQ. Then of course, along with that, the maintenance of the equipment. You know, all the people, that if there's a maintenance issue, then definitely you are having a lot of problems in, you know, deviation and all that. He's the expert, he is going to give you all those things. But these are a headache. So at least we are getting very good service. After sales service is also very important in that. So I can say that we have been very happy. We have a lot of equipments which are working, and I wish Mehul Bhai and his team a great success. I'm sure he is going to make a big leap in this area, as we have already seen. Whenever I get some customers or some dignitaries from outside, when they see our equipments, the first thing what they say, it is imported. Then we say, no, 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 it is from Gujarat only. So I think this is a good sign that people are comparing you with the imported equipments, whatever. I don't say imported equipments are very good, but I think you have done a good thing, and I congratulate you, and I wish you all the best for this seminar, and I am sure you will grow much more than this. In the end, I will say best of luck to all the Kesar family and Mehul Bhai. Thank you.
Thank you very much, Shived, for such lovely remarks for the company. Friends, our second guest of honor is today Dr. Viranchi Shah, promoter of Saga Life Sciences Limited and companies under the Saga Group. A pharmacist and techno-entrepreneur, Dr. Shah leads Saga Group in manufacturing high-quality, affordable medicines. As the immediate past National President of the Indian Drug Manufacturers Association, the world's largest association of pharma manufacturers. I request Mr. Jagdish Patel, Managing Director at Kesar Group, to kindly felicitate Dr. Viranchi Shah. Also, as the sequence goes, I request you to please say a few words for this introduction. Thank you so much.
Good evening, dignitaries on the dais, off the dais, and friends from the industry. On the onset, I would like to congratulate Kesar Control Systems and Mehul Bhai for completing 16 long years of service to the Indian pharmaceutical industry and the good work that he has been doing in this field. I echo the sentiments that have been shared by Sudhir Bhai. We too are very satisfied users of products from Kesar. We have stability chambers from them in our EU GMP facility, and we are more than happy. In fact, for our R&D centers, we are already in negotiations with them for some more units. But that is not why I am here. More importantly for me, we also have Dr. SARANJIT Singh, and I have been knowing him for some time, and the good work that he has been doing in the field of pharmaceuticals. Friends, stability testing is gaining very critical importance in pharmaceuticals. On the 30th and 31st of last month, on July, we had a global drug regulatory conclave in New Delhi, where under the aegis of the Ministry of Commerce and Ministry of Health of the Government of India, we had about 60 global regulators, including the US FDA, the UK MHRA, Japan FDA, and several other regulators. And I, being the Committee of Administration of Pharmaxil, we were the organizers of that interaction. We had the state regulators, the central regulator, and one of the most important message that has gone out from India in this conclave was that India is in for one world, one quality. And when we say one world, one quality, we must understand that gradually we will be aligning to global standards. So while there is a lot of debate going on what should we do about Schedule M and whether it is adaptable by industry or not, be ready that in the coming decade or so, we would be a part of PICS. In fact, as far as the ICH is concerned, because ICH has a lot to do with the stability study which is going to be discussed today, we should be applying for membership as early as end of 27 or early 28, which means that especially in the field of, for example, stability testing, a lot of progress is going to be made in terms of India's commitment to global harmonization. Those who are in analysis, you must have understood, you must have realized that even the Indian pharmacopoeia, because we are a part of the global discussion group along with the USP and the BP, the Indian pharmacopoeia is slowly getting aligned with the BP and USP. So these are something that is written on wall, and for all of us who belong from the industry, it is very important for us to be early adopters, so that the day when these changes are there in black and white, we don't have to run here and there to see how we can buy more time. And when as a nation we project ourselves as global leaders, third largest manufacturer, etc., it is also incumbent upon us to ensure that we align to global standards. In this context, I sincerely appreciate Mehul Bhai and the team Kesar for bringing discussion on this very important group, which I am sure will help you understand the nuances of stability testing and how your individual companies can do well in this area. I compliment once again Kesar Control for the good work that they do, and I wish a wonderful engagement to all of you. Thank you.
Thank you very much, Dr. Viranchi Shah, for such encouraging words. Our next guest of honor, ladies and gentlemen, is Dr. Srinivas Savle, CEO of the AIC LMCP Foundation at LM College of Pharmacy, Ahmedabad, and Executive Committee member of the foundation. Dr. Savle holds a PhD and has over 22 years experience across drug research, compliance, CRO setups, and academia. His expertise includes compliance, data integrity, lab automation, electronic workflows, and more. I request Mr. Kalpen Patel, CEO of Kesar Group, to kindly felicitate Dr. Srinivas Savle. Also request you, sir, if you could come up and share your thoughts with us. Thank you.
Good evening, everyone. Actually, I'm feeling a little embarrassed sitting with some dignitaries like Dr. SARANJIT Singh, Sudhir Bhai, and Viranchi, and sharing dais with them. This is all because Mehul insisted that I should be here and represent LM College of Pharmacy. Now, I share the sentiments from the academic settings, like the incubation center that I take care of. Basically, is trying to foster entrepreneurship in pharma and healthcare sector. And we also have sophisticated labs for helping the startups to actually try and test their products and take their technologies forward. And you name the equipment, and all the Kesar equipments are there at LMCP and AIC LMCP Foundation. So right from incubators, BOD incubator, autoclave, stability chamber, freezers, all range of temperature and humidity equipments that we have. And Mehul Bhai was one of those strong supporters for this institute. And one particular aspect that he has seen, that the instrument should not be down for more than 24 hours. Any instrument in academic institute which is having downtime less than 24 hours is never heard of. So thanks Mehul Bhai for all this support. I am sure that in the capacity of auditing some of the pharma and CRO settings also, I have seen Kesar instruments everywhere, and that has never been an area of concern as far as qualification, calibrations, and routine maintenance of these equipments, which are critical. Especially you talk about pharmacies of CROs. Kesar stability chambers are actually doing a wonderful job, because these are 24/7 continuously running equipments with continuous monitoring of data, and which is heavily scrutinized by regulators. So you talk about FDAs or EMAs or any other regulatory agencies. I think this is one soft spot for them to really look at the quality of operations and send the quality focus of the founders of those companies or management of that company. So when it comes to stability, Dr. Saranjit Singh is one of the global thought leaders when it comes to even framing of the guidelines. He was involved earlier, and I'm sure it's a rich pool of knowledge that he is going to share with us on stability. More so, we are talking about now novel modalities that are coming. So it's not only the small molecule space, we are also working in now biologics, biosimilars, we are talking about cell therapies and all, where again the shelf life and stability becomes so critical. I'm sure we'll learn so many aspects around it from Professor Saranjit Singh. And at this point, I congratulate Kesar Group for the fantastic support they are providing to their customers. And I especially thank Mehul Bhai for this opportunity. Thank you.
Thank you so much, Mr. Sable, for such encouraging and motivating words. It is now my honor, ladies and gentlemen, and I was looking forward to this moment, quite proud of it, to introduce to all of you today's keynote speaker, Dr. Saranjit Singh, former Acting Director, Dean, and Head of the Department of Pharmaceutical Analysis at NIPER, SAS Nagar, where he served from 1994 till his superannuation October 2021. University gold medalist from Punjab University, Dr. Singh brings over 40 years of teaching and research excellence. He's an internationally recognized authority in drug stability testing, degradation, chemistry, and impurity profiling. His extensive body of work includes approximately 230 research papers, articles, and book chapters, one patent, and significant research that has influenced Indian and international pharmacopoeias. Dr. Singh has served as a temporary advisor to the World Health Organization and as a member of the expert advisory panel on international pharmacopoeia. A dedicated mentor who has guided 135 masters and 15 PhD students, executed 100 sponsored projects from the industry. We are so proud of you, sir, to be here. Thank you so much. I request Mr. Chetan Mistry, Vice President of Kesar Group, to felicitate Dr. Saranjit Singh. वो शुरू वाली तालियां कहाँ गई? Thank you. Thank you very much. Friends, quality, they say, is never an accident. Quality never happens as an accident. It is always the result of intelligent effort. In pharmaceutical manufacturing, where products directly impact human lives, maintaining stability and compliance is a fundamental responsibility. To share insight into Kesar Group's journey and technical direction, please welcome, with a huge round of applause, Mr. Sandeep Desai, Chief Sales Head of Kesar Group, for his presentation. Huge round of applause bola tha na? Thank you, gentlemen, for joining us here. We look forward to your services and the session with you, sir.
Hello. Good evening. So, good evening, everyone. Myself, Sandeep Desai. I'm heading the sales activity with the Kesar Control Systems, almost since 10 years. First of all, I would like to very warm welcome you all, our respected guests, clients, and consultants, and industry professionals. Also, on behalf of our entire Kesar Group family, I would like to thank you for taking the time to join us today. As we know that today's seminar is focused on the most important aspects of the pharmaceutical studies, product safety, quality, and regulatory compliance. But before moving with this technical session, I would like to introduce our Kesar journey with you all. So, as we know that every successful organization has some story, and our begins with solving the customer's problems. As our technical director, Mr. Mehul Patel, he started his career in 2001 as a technical service engineer from Mumbai with the same company, similar, like they are specializing in laboratory equipments. So after working closely with the pharma industry, he gained so much practical knowledge about the customer requirements and industry challenges. So in 2009, he started his own venture with a very small team. And we can say that that small team that at that time that was started now is a very trusted brand in pharmaceutical laboratory equipments. Today that we are proud that how far we have come. We have more than 220 dedicated employees with the Kesar team. Also, we have a global presence, more than 65 countries. And we have international as well as domestic service supports are available wherever our customers are located. But I feel that more than these numbers, what matter most to us is the trust that customer have placed in Kesar Group. So I'm really thankful for that. As we have that completed successfully thousands of installations through pharmaceutical industries. Also, we do have a service supports wherever our customers are located. And we believe that quality is never a choice. It is our responsibility. Okay. So we believe that whatever the machines we manufacture and we dispatch from our factory, it should meet international pharmaceutical standards. So moving forward, as you can see this chart, it reflects our journey, past journey over the several years. You can see so every milestone that reflects our customer confidence and continuous innovation, and we can say completely hard work of our entire team, right? So our goal is not to simply achieve the highest turnover, but our real goal is to provide the reliable solution to the pharmaceutical companies so they can maintain their standards of quality. At Kesar Group, we believe that the technology should ultimately serve the humanity. So whatever the medicines reaches to the patient, it should be properly tested and with the proper storage conditions, right? So that's why whatever the chambers we manufacture, that carries a completely greater responsibility with this. Now talking about our production facility, we do have that modern production technology
We do have in-house CNC machines, as well as turret punch machines, laser cutting and puff filling machines. All the machineries we do have in-house. So whatever the quality, we can have a look out on this. So now moving forward, with the Kesar control systems, we have another group that is called the Kesar testing services, right? This is completely separate group, and we do provide the services for the calibration, validation, testings, and all those accreditation laboratory. In this lab, we have a scope of this: thermal, pressure, electrotechnical, dimensional, mass, volume. Also, we do calibration, HVAC calibration and clean room validation. All the facilities we provide with this accreditation lab. And also we can say this is the backbone of our Kesar control, because whatever the machines and we manufacture, that components what we do with the calibration with our NABL lab. So we don't rely on any outsourcing. Now, I would like to highlight our premier flagship innovation, that is eco-intelligent model. So, before 5 or 7 years ago, we have launched our EcoSmart walk-in stability chamber. But while in that technology, we are not using the boiler heaters, or at that time we were using the ultrasonic transducers. And earlier in traditional technology, our compressor was like continuous working. While in EcoSmart, it is on-off kind of systems. So, but there were only one limitations. Below the 8,000 liter, we cannot go with that technology. But after our team hard work, we have launched this eco-intelligent stability that starts from 400 liter only. So that will be ultimately beneficial to our customers. That will save your 60% overall energy and 70% water. You can see the comparison. It is not a heater based, it is ultrasonic, and the humidity and temperature response is very instant in this technology. And these are our products range, what we are offering. EcoSmart stability chambers, photo stability chambers, and we do have also micro setups for BOD incubators, autoclaves, ovens, muffle furnace, dehumidifiers. Also, we do have some certifications, that is compliance to European standard, CE, ISO. So we also, our quality recognized by internationally accepted certifications. So we have a manufacturing facility located in Vatwa, Ahmedabad. We have a 2 manufacturing unit, unit 1, where there we are manufacturing stability chambers, incubators and all. And remaining unit 2, we started our clean room equipments, that is the pass box, biosafety cabinets, LF, RLF, etc. So at last, I just want to say that when you partner with the Kesar group, so you receive the higher precision engineering and lower operating costs and completely peace of mind audit. Thank you, and I wish you all the very successful seminar. Thank you once again. Now, I would like to display our corporate video.
Less than 100% is nothing. That belief has kept us moving forward, giving our best and drawing the best out of every resource. This is Kesar Control Systems Private Limited. Founded in Ahmedabad, Gujarat, Kesar Control Systems began as a service center in 2007 and scaled into full manufacturing by 2009. Today we operate 2 units in GIDC Vatva, designing and manufacturing pharmaceutical, and laboratory equipment recognized for durability, unique design and smooth operation. Backed by ISO 9001:2015 and CE certification. Meet the team, the backbone of our spirit and pride. Over 200 professionals qualified to take on the toughest assignments. Committed to zero-error delivery. Our vision defines the goal. Our optimism defines progress. Our ethos is simple. Good begets good. We give our best to customers, vendors, and every stakeholder, leaving nothing to chance. By streamlining every stage of manufacturing, we stay ahead of the competition, offering a one-stop solution backed by annual maintenance contracts and 24/7, 365-day service support. Every Kesar equipment is fabricated on fully automatic CNC and turret punch machines with precision bending systems, manufactured to GMP and ICH guidelines and conforming to MCA and US FDA requirements. Our range speaks for itself. The Eco Intelligence and Eco Smart Stability Chamber saves up to 60 to 70% power and 80 to 90% water over traditional type stability chamber. Alongside it, stability and humidity chambers, photo stability and dual chambers, BOD and bacteriological incubators, cold chambers, hot air and vacuum ovens, muffle furnaces, deep freezers, ultra-low freezers and vertical autoclaves. A clean room equipment line, laminar airflow units, pass boxes, biosafety cabinets and air showers extends our expertise into controlled manufacturing environments. We operate our own NABL accredited calibration laboratory, offering periodic third-party calibration as part of our service arsenal. Every equipment ships with a full documentation package. Design, operational, installation and performance qualifications, along with maintenance and operating procedures. Our quality team inspects every unit before dispatch. Kesar Control Systems equipment is trusted in more than 65 countries, with over 7,500 units supplied worldwide. We serve some of the biggest names in pharmaceuticals and research. Across chemicals, nutraceuticals, biotechnology, API, medical devices and cosmetics. Customers are our highest priority. In that spirit, Kesar Control Systems works every day to become an indispensable partner to the pharmaceutical industry, because every employee has a vital part to play in one goal: to give our best and never settle for anything less than 100%.
Myself, I am DG in Quality Control from Pvt.Ltd. We are using Kesar Control equipments, stability chamber, humidity chamber, and performance-wise is very good. Thank you.
Also, I would like to add that we have demonstrated our eco-intelligent model over here. So if anyone interested for it, so they can reach out to our team. Thank you. Yeah, after the seminar. Thank you.
Thank you, Mr. Sandeep, for your presentation and for a lovely audio-visual. Hope all of you liked it. Right, so we move on. Short note, delegates, please take a moment to complete the feedback forms, as I was saying at the beginning, which are in your welcome kits, and submit it before leaving. That'll be nice. Your inputs will be greatly appreciated. So, as we know, today's main session addresses a critical pillar of pharmaceutical quality assurance: pharmaceutical stability testing storage facilities. Over the next few hours, we will explore scientifically designed storage environments, regulatory expectations, stability protocols, risk management, and best operational practices. It is an absolute privilege, like I said, to hand over the stage to our eminent keynote speaker for the evening. Ladies and gentlemen, please join me in welcoming Professor SARANJIT Singh. Sir. Now the stage is finally yours, sir. Sat Sri Akal.
Okay, friends, very good evening. I don't see anybody in the hall because I didn't get reply on my good evening wishes. There's nobody in the hall, I can't see anybody. Very good evening. Yeah, that's the spirit. I think I should repeat 3 times. In Punjab, when we say Jo Bole So Nihal, we say 5 times. So, very good evening. Very good evening. Very good evening. And finally, very good evening. Okay. Now, say, I am going to be with you for the next almost 3 hours, not 3 years. I don't want to test your shelf life. So for 3 hours I am going to be with you. And when I start, I always say that you're here for a purpose. Purpose is why we are here. We need to understand. The reason is that this topic under which I am going to have discussion today has never, no seminar has been held in the country earlier to this. Believe me, this is my live 626th lecture. Never on this topic. The problem with if you are working in exclusive area, which I have followed from the very early days of my research career and the professional career, working in the area of stability testing, the problem is that if you are so closely associated with the topic, people will tend to change your name also. I am known in the country more as a Dr. Stability Singh instead of Dr. SARANJIT Singh. Now that is also a situation that can happen to you. So these 3 hours, now I am going to show you, treat it as if you are sitting in a film theater and looking into a favorite film. Why say so? Once I was at Sipla, the session started at 9 o'clock in the morning, finished at 10 o'clock in the evening. Not a single person went out of the hall. And at the dinner time, after 10 o'clock, somebody comes and tells me, I was a fool all the lifetime that I was looking into Amitabh Bachchan as a star, but real professional and technical star is before us today. That also the platform, and I was very funny sitting over here, trying to come and sit over here, the press people will pull me out. Give a bite, give a bite. I said, what is this going on? An academic person in the midst of technocrats, professionals, is getting that honor, and this is all credit to Kesar Group. Mr. Mohali Patel, who really has organized this event, big event, and we are counting the number of registrations. Where to close? He was in discussion with me where to close. 200, 300, how much arrangement? Then he shifted the hotel, bigger number, and today he says that this seminar is being, the gathering and the registration number is 600 plus today. Now this response to a technical seminar is, I will say, so encouraging for all of us. The onus comes on me how to keep you bound to your chairs for 3 hours. So my request is that we have volunteers, including Mr. Mohali Patel standing over here, the CEO of the company, that he will not allow you to get out of the room while I am there speaking to you. He has invested a lot in organization for planning going on for a very long time. And the onus comes to me to keep you bound to your chairs. Interestingly, I will use my whole life experience to keep you in your chairs. Only for bio break you can, or small request, nature's call, you can move out and come back quickly. The reason is, pharma industry in India is so fragmented from small scale, medium scale, large scale, Indian multinationals, foreign multinationals. And the practices of stability testing vary very widely. Small scale wants a stability chamber as a showpiece, not to waste electricity on it. In the morning time it will be running, no samples added, just dummy samples are put over there. That is one practice. If you go higher, you'll find some small group activity. Every fresher which is employed in the company is posted in the stability area because it's a mundane, routine type of job. But as you go to the bigger companies, where you find 3,000, 4,000, 5,000 HPLC working 3 shifts a day, they have extensive areas dedicated to stability storage facilities. Now regulations are same, inspectors are same. Now whose product we should consider stable or unstable? Now this is a big trauma for any country, and same trauma is of the regulatory officials who come over to inspect your facilities. But there are, I should not shy to say that, there are regulators who are happy with the Diwali cash gifts, and they will not tend to even visit your facility to look into what is going on with respect to GMP and other operations. Long back, we had a few manufacturers that had taken initiative to launch stability chambers in this country. Because I was from the, I will say, 80s, I was given a problem. My boss, late Professor S.K. Baweja, professor at Punjab University, he was a person who kept interest in stability testing, science. He pushed me into this. And at that time, my concern was that there should be facilities made within the country so that our industry growth, parallel to our industry growth, there should be technologies and the instrument equipment available manufactured in our country. So one or two of these manufacturers who were leading in the country, they got in touch with me because their chambers were initially very primitive designs. They were getting rusted. Now what you see, steel chambers and beautiful GMP models, they were not existent at that time. There was no technology even for welding steel in a wider sector. Few companies had that, but not at a wider scale. Looking into that there is a need of development, they used to come to where I was staying in Delhi or hotels, and they used to come and sit with me, spend multiple hours with me, how to really bring up those. And Dr. Mehul Patel, Mr. Mehul Patel comes out of that one company and who started Kesar zone, based on the experience with that company. When I came and joined NIPER in Mohali, the institute started with my joining on 1st August 1994, and we had to organize all the facilities and equipment. So I thought that I should import, our emphasis was importing the best equipment to do the best research, possible research. So I bought these chambers which are shown over here. They were from Binder, Germany. Now, I got 4 of them for my lab for different storage conditions. And believe me, when they came, like a typical, I can give you an example of a light chamber. They were marking as a plant growth chamber over there in Europe. They sent the same with the lights on the door. When they came to me, I said, what is this? They showed me pharmaceutical stability chamber with lights on the door, how I put my tablets and expose my tablets to the light. Very interesting. Then I had to use a thermocol sheet. So use the thermocol sheet, put pins over there and hold a tablet inside 4 pins. That way we used to expose. But I really felt very bad, and the person who used to come as a salesperson to me, I said, nothing doing, I need another chamber. He said, but we have transported it, spent a lot of money on it, we cannot take back and give you a replacement. I said, fine, you send me. Then I listed around 20 flaws to that salesperson, that these are the flaws with your stability chambers. Critically studied each of them. Believe me, Mr. Binder himself, whose name company was, he invited me to Germany, that you come over and discuss with my team what are the problems with these stability chambers and how we can be the best in the world and manufacture our system which can satisfy pharmaceutical stability testing. Now, I landed at Stuttgart in Germany, and this plant was in Tuttlingen, which was around 100 kilometers away. And the person had come to pick me up. While we were driving, he was driving me to Tuttlingen. On the way was Mercedes factory, which manufactures Mercedes cars. I really was thinking from my side, new professor at NIPER, corner room in one of the blocks, and the precision engineering in Mercedes cars, and I am being invited to discuss about stability chambers and the precision that can be built in the stability chambers. Laughing at myself internally, that how come it has happened that will be able to convince, and what I was able to convince them is that pharmaceutical stability testing requires IQ, OQ, PQ, which they never had documentation. So very small tips. I spent whole day over there. So the question comes over here is, the lesson which I am trying to tell you, that we have to go a little deeper, whether this device is standing over there. Yesterday, very funny thing happened. I landed and directly went to the company, factory. Very interesting. To Kesar control system, Mr. Mohali Patel, despite being all the busy in organizing this whole, was waiting for me. He asked me to have a round. Mr. Dinesh, my friend who accompanied me from Chandigarh, technical, excellent hand on the north region, he was also moving. Looking into everything, you might have seen in video that they are filling puff machine, puff insulation in the 2 plates. Just taking a round of it, a keen observation, I asked, how are you assuring and satisfying me that the puff, that lining you are putting in between 2 plates, outer plates, is uniformly spread without holes? Because if there is a hole, there will be a tendency to equilibrate with the outside temperature. So how do you really prove me this? Have you any ultrasonic system to prove that there are no holes left when it's a chemical reaction leading to swelling and formation of puff. So question comes over here is, when we are dealing with an equipment, how much curiosity in everything that goes into construction and working comes to your mind. That is very, very important, my friends. And that only will take India forward, the country forward. Until unless you have that, and that criticality of mind, observation brings me here. I am academic. I have taught something different to my students all the lifetime. But what brings me before this gathering, esteemed gathering of industry friends, technology friends, manufacturers, consultants, is that criticality of mine. And yesterday I challenged them. This chamber you have said, it's your innovation of last 5 years. Ecosystem. You have brought some new technologies. What is your next plan for the next 5 years, or what new innovation you are going to do? Unless we are innovative minds, think innovatively, we cannot take our country forward, please. And more important is culture of sincerity and integrity that we are missing. I was at London Street once. Believe me, by nature Punjabi. And I never saw that at the zebra lines there is a pole which gives a green or red light that you can only cross on foot. After seeing that, it is green, because we see only in the vehicle that is the red light and green light, not for when you are walking on the zebra line. Believe me, such was the culture and discipline that I am in the middle of zebra line and huge number of cars are speeding up. And I am in the middle of that road on the zebra line, wrongly to me. But every car could have crashed each other because they stopped suddenly. They saved me, ignoring their safety. And it was a very big lesson for me. And nothing like this, that in Punjab what we do is, we'll get out. Oh. 2 bad words, 3 bad words, first thing which will speak to that person, you don't know that this is the green light for us, not for you. That cultural change, which is very much regional, I can say. Some places there is a very strong culture in India, some places it's a totally bad words that are spoken on some thing which may be unintentional. So we have to really develop that culture of integrity, culture of truthfulness. What to say? You will say you are praising a lot about yourself. But if I am known worldwide today on stability testing, it's a sincere effort. Spending time in the institute from 10 in the morning or 9 in the morning to 8:30 after spending whole day with the students. Brightest students. And thank to them that their effort has brought me over here, because they have been contributing a lot in the terms of what I am today. Now question is, this is going to be a very boring topic otherwise. Storage facilities, a few chambers. But what I want to highlight in these 3 hours to you, how important they are, what are the international regulations that are emerging. Very small point can lead to a total change. Now, I will come to slides a little later. What is bringing a change all over the thinking in the pharmaceutical industry worldwide? Let me discuss few minutes, 10 minutes on that. The whole concept which is going on in industry is build quality, don't test quality. Quality by design. Now what we do today is we build billions of pieces of pharmaceuticals in pharma factory. Cumulative. Billions of pieces of tablets, capsules. What we do is, when I am now talking as a consumer of medicines, You also, anybody in the hall, anybody among 500 people over here, please raise hand. If someone of you has not taken a medicine till now in life, in lifetime, let me know. Any hand raised who has not taken a pill in his lifetime? You youngsters are very healthy people. You should not have taken medicine. But I see no hands raised. We are all consumers of drugs. Yes or no? Now, if we are consumer of drug, how you are putting a faith in your tablet that goes to your mouth? That's a very big question. Am I taking the right pill? Will it act? Yes or no? It's a faith. But faith is dependent on, I told you, the industry practices that very variable, fragmented industry with very variable practice, whom to believe, whom not to believe, that if the pill I am taking will act or not. A simple example, soft gel capsule, hard gel capsules. Gelatin is a protein, gets cross-linked. On exposure to high temperature, humidity, gets cross-linked. Then it is also cross-linked through aldehyde impurities present in any of the excipients that goes into making a formulation that goes into soft gel capsule. And cross-linking means, protein cross-linking means that it will not dissolve at all. Sometimes you will not come to know that you have taken a pill, capsule you have taken, it is over cross-linked, beautiful capsules, shining capsules, but it has undergone cross-linking, and that whole capsule goes out in the feces in the morning undissolved. Big problem of Indian industry. You will never come to know. You don't look back into the toilet to find out that the formulation was there or not. It's a practical thing. So this is a deep science, making pharmaceutical, producing pharmaceuticals of million pieces, billion pieces. They are going to affect your health directly. Our health, my family's health. It's a big question. Now, building quality by design, building quality into the product, not testing quality. Can you believe a situation where I don't test product at all for the quality? I design quality into the product and every tablet has the same 100% drug strength. Can you think of a situation with a machine, automatic machine punching out 5.5 lakh tablets per hour, and each tablet has a. Now, very interesting, further, among you three ladies, how many, all of you had traveled by air? Yes or no? Very simple question. All of you must have traveled by air. You have felt safe in the aircraft or not? No? Truly not? Okay. These are three ladies. Let me ask three gents. How about you people? Did you feel safe when you had traveled by air? You had confidence in the plane and the pilot? Yes or no?
We have to trust God.
Trust God? Wow. And here in your city. Okay. Now, have you ever heard? Now, that moment, if you look into the radar on your systems, you can see at this moment almost 10 to 15,000 flights in the air at this moment. Okay? And millions of travelers are flying from one place to another. Odd situation of a plane crash can happen anytime. But have you heard about that there has been testing done with a full load of you people, and pilot speaks to you, today we are going to do a crash test on this plane with full loaded with you people. Has it ever happened? Yes or no? Okay, I ask you a question. Are you married? No question to you. You are married? You have a child? Small? No, we don't need a mic at this moment. If I have to do clinical trial on your newborn child of 0 day, 1 day, because sometimes they also need drugs due to one reason or another, well, as a mother, you would allow me to do that? No. Then how do I translate the dose of drug for a newborn if your mothers are not ethically allowing me to do that clinical trial? Same thing happens to the flight. No one allows me to allow a crash test to happen. And do you know, any guess from this audience to tell me how many are components in a plane? A plane is made of how many components? Had you been critical, and you might have seen a pamphlet in front of your seat which had this number displayed in Vistara. How many components are there in a plane? Any guess? Any guess behind? In Vistara, yes, what is the guess? 2000. 82. 6, 4, 64,000. From where this number has come? कोई और दे दो rough estimate. I was myself surprised. 64,000 components, what he is saying. I was myself surprised. I read that the card over there, it says this plane has 20 lakh components. Why I'm stressing on this so much? 20 lakh component plane without a crash test is flying you safely from one place to another, with the life of pilot and the staff also involved. Yes or no? How they can do that, build a plane with 20 lakh components, and it happened in Nagpur, I tell you. There was a skidding of the plane from the runway, and there's a big splash news. By chance, I was there at the same airport in a week period. I asked the airport manager, how this happened? He says it was pilot error. Judgment that he could not stop the plane, slow down the plane before the edge of the runway, that it went into the grass. I asked her, is it normal for the plane to happen? He said, no. Airplanes are the safest mode of travel on earth. Otherwise, they are so well computerized and constructed, nothing, no error can come so easily. That is a confidence when these companies build up the planes in which you travel safely. Millions of passengers travel in certain period of time. Do we have the same confidence in our 10 component tablet, which we build up billions of pieces? And it's going to affect billions of patients. Yes, no. क्या यार डांट खाओगे मेरे से? How they build up. I tell you the story of my childhood. My paternal aunt gifted, she was in US, she came for a visit to us, She gifted a Rado watch to my father. And as it would happen, he was a Gandhian wearing khadi. He never was fancy to having a jewel-loaded watch with automatic watch with gold plating. He never wanted to display that. So it was lying over there. When we started growing up, we also saw that something is lying and watch. And when we wanted to be a show to peers and friends that we want and we will start him borrowing that watch from father. And he happily said, okay, fine, take care of it. Old people must be knowing, now also Rado is famous, but it was very old time. Slowly, as the kids would do, something went wrong with that watch, and automatic behavior, it stopped working practically. I went to a shopkeeper, the mender of the watches, a small box type, the person sitting behind it. He told me, he looked into the hole, opened the back cover and tried to find out the fault. He said, try to open the small boxes for components, but he could not find. It will take 1 week for me to repair this watch. I said, so many components are there, why don't you put one and put in order? My father will really bash me up for pushing this watch into that bad state that is not working. When he finds that, I'm going to get a bashing today. I tried to ask him, what is the reason you are unable to repair it? He said, Papa, this watch, each component is so greatly well polished that it is almost all the gears are friction free. And that's why it maintains the time which Mr. Mehul team was exposing over here, their NABL lab which they have in the company. What is a job? Job is atomic clock which drifts a few seconds over the years. And our watch drifts minutes in a month or 2 months or 6 months. So they have built so much of the precision through making it totally frictionless. So build quality, not test quality. This is a concept which is coming to pharma. Quality by design. Build quality, don't test quality. So friends, I can continue talking 3 hours, then I will not be able to touch my slides. But this culture of quality. Now, it's not that. Tomorrow you may say, my regulatory people are, I will say corrupt or whatever, you can label them. They don't allow to us to prosper, they don't allow, but it is your hidden. Whether I want to be part of that game or I want to be different. If you can be different, then you will come and stand over here where I am today. That is what India needs. Thank you so much for listening up to this point. Now I go to the next slide. Now, what I put over here, mutual learning session today. You are all welcome. What is mutual learning? I have an academic with a zero industry experience, standing to speak something which is very close to industry, a requirement of industry, by mandate on industry. Now I have to learn from you and be open to ask any question anytime. I'm a teacher, I can take question anytime you can intervene me. We have volunteers, she is standing, you can take a chair in between, don't worry, I will ask you to get up and hand over the mic, please stand, sit down, be comfortable. I know you have been given this mandate, but kabhi liberty le lete hain, baith jao. Now, issue is, I want every query of every person answered If I can answer, fine, but if I am unable to, I don't claim that I will be able to answer every, but any other expert among seniors sitting over here who are handling the stability departments in industry, if they can intervene, I will most welcome. So let us have a very interactive session. Now, here we need to understand another thing. I am differentiating over here the difference between academia and the industry. What is the difference? In academia, on the left side, we built experts in their own fields. Like Niper allowed me a platform to build Saranjit Singh on stability testing, a pillar in stability testing known world over, as they have introduced. I may not say myself that I am known world over, but others are saying you are known world over. Fine. So we built parallel pillars. Somebody is a very good, excellent medicine chemist. Some is a pharmacologist of repute. So they are known world over. But in industry, there's a pyramidal structure. Everybody, here what I see the star on the top is the product you have to launch in the market. Successful product you have to launch in the market. Now, if this has to be launched in the market, against the competition from all other manufacturers, you have brought some new technology. Everybody has to function in unison to launch that product and to make it successful in the market. So there has to be a common understanding within a company to grow forward, to have much speedier implementation of the product and put the product in the market. So it's a totally, entirely different structure. You have to understand. But there is no, today we were in discussion with, I went to meet Dr. Shrotriya, who is an industry veteran, Dr. P.G. Shrotriya. He was telling, in industry without academia, criticality of academia, industry is not successful. So there has to be a very close industry-academia relationship to succeed. Starting very simply, what is stability? You talk stable government, collapse karjegi if they are not stable, it will collapse, change of government will happen. So here, simple definition, if I go look into Google and ask for the definition of stability, means the state of being stable. What is drug and product stability? The extent to which a drug substance or product retains within specified limits and throughout its period of storage and use the same properties and characteristics that it possessed at the time of its manufacture. So what we want is that the product, before I pop a pill, it should remain, it have its characteristics within that specifications which are set by either regulatory pharmacopoeia or in-house. What is stability testing? Set of studies utilized to determine if drug substance or drug product is altered over time by various environmental factors such as light, temperature and humidity. Your product, when it is on the chemist shelf, or the worst situation is your pharmacy box at home, drug box at home. Please, all of you, while enjoying your refreshment, bring into your mind how you handle the drugs at your home. What respect you give to your drugs packages at your home. You have everything in your box, yes or no? And you shuffle out to find out a drug you want, yes or no? The product you want, you shuffle out. What you are doing in the whole process is thinning the aluminum layer, package layer. And in India, very funny thing happens is that doctor writes the prescription, you will take a few more drug tablets or capsules out of the whole strip, and we are so poor as a society. If I say, madam, what is your annual earning of your family? I will say 1 CR. Still, when you go to a chemist shop, you will ask, Nick, give me 4 tablets only. Yes or no? You do that. Next time I will pay and get more of it because I don't have the money enough in my pocket at this moment. It's a household way of doing things. Now, the retentivity, because what happens is that you have taken 4 tablets, used 4 tablets, you are not cured. You will go to the doctor, he will change the brand of some drug. So what will happen? That the residual part of the strip will lie in your home for as long as you need next time. Wanted or unwanted? Yes or no? Is it a reality? Yes or no? And for the years you have kept it, it is getting shuffled with other formulations in that drug box, packaging in that drug box. You are thinning. And the effect of environment, effect of humidity, light, everything can affect the stability of the product. So what we are talking about stability chamber, photostability chamber is meant to test your quality of the drug and the change that can happen over the period. But mind it, the stability testing we do in a chamber, we don't have any experiment of the shuffling you do at homes. We don't have any experiment developed to check for the shuffling effect on the product quality and stability. What is pharmaceutical stability testing storage facilities? Try to read it. While you are having your refreshment, try to read which are you are talking today. Because when you read yourself, if you are unable to understand, then ask me, other definition is very clear. So basically, you can see the photographs over here. This is what we are talking about. These are the chambers, special purpose chamber, which retains certain level of temperature and humidity. And then we have photostability chambers, which gives you test for photostability, the light effect on the products. Any question? Today, my request is the question should be limited to the topic, not general stability. Now, the storage facilities is a very small component of the whole stability testing program. You see the titles over here. Read out the inference. This is whole stability testing program which goes into industry operations. So first is stability department organization. Then we have stability study design, which should be robust. Standard operating procedure, complete set. There may be hundreds of SOPs that may be needed in the stability testing laboratory. Then we have sample management, where to keep the samples, the lots, how many samples are to be kept, what is the number of tablet lot which have to go into stability chamber, because all the facilities and the spaces you create, the volume you create is dependent upon these facilities requirements. Then sample storage chambers, numbers, capacities, qualification. I will go more detail into this: storage condition, location monitoring, time point scheduling and sample pooling management, testing workload management and risk management. Then sample collection and analysis, all the functions that goes after you have stored the samples in the chamber. Data analysis, review and approvals. Now you have to determine the shelf life expiry date from the data you get. And then predictive software are there these days where they are used. And then regulatory reporting, which should satisfy the regulators worldwide. Here is only the part that is the topic of today's discussion. Now, very interesting is, all the guidelines suggest that we should do the calculation and determine shelf life, expiry date based on 3 batches, initial production batches. Now, I have the 3 batch data. Here I am talking of that I have 5 points for each batch. I plot over here. Here it is time, and this is the intercept, means the value, SA value versus time plot. And what can have deviation or out of specification results or out of trend results, that I am trying to put over here. If the intercept and the slope, they are identical, intercept is same, slope is same, all the 3 batches are showing same, it is ideal situation. I can easily take average and determine the shelf life based on the procedure that has been prescribed. If the intercepts are different, slopes are same, what happens? It means the initial drug strength in each batch is different. That's why the kinetics is same, but initial strength of drug, let us say instead of 100 it is 105 or it is 95 in one batch. Initial drug assay. Slopes are same, still I can pool. If there are storage factors interplay, that in the stability chamber, in one location, the temperature humidity is differential than other location, the slopes would not be same in the 3 batches. There is going to be a situation where you cannot pool the data, and here in case of disagreement, stability estimates to be made on the worst batch, on the single worst batch. And if slope and your intercepts are both varying, then it's a further crisis situation. It means drug strength is also varying, as well as storage conditions are also varying. Again, you have to go for the worst batch in the right 2 cases. That's why so much big emphasis is there on the storage chamber and the preservation of condition inside that chamber. You understand what I am trying to say? So what has happened is, internationally, there are international guidelines to help the industry to run the stability program effectively, efficiently. The guidelines have come, which are regional, international guidelines, regional guidelines, national guidelines. But ICH and WHO guidance documents, they exert maximum influence. Today also, in the seminar, they were saying ICH, ICH, ICH. So these documents are considered to be the standardized documents which have an applicability all over the world. Now, believe me, in India, we don't have our own stability testing guideline. We have no letter issued by CDSCO which says India has adopted ICH or WHO guidelines, stability testing guidelines. And we call ourselves as the pharmacy of the world. Because the good part is India is exporting the drugs to 200 plus countries. They are asking for ICH guidelines. You should follow the ICH guidelines. And without a regulatory mandate, our industry is following the ICH or WHO guideline, whichever is the mandate. But in India, we don't have any formal recognized document which say that we have adopted ICH or WHO guideline. A huge weakness in our regulatory system. Now, these are the ICH guidelines, which I want to talk about first. Now, why ICH was born? What is ICH? International Conference on Harmonization. What was the purpose of it? purpose was that there are 3 regions of the world, US, Europe and Japan. They are the biggest innovator of new drugs. Combined, US, Europe and Japan. Biggest innovators, biggest producers of new drugs and biggest consumers of new drugs. Can you really, if I give you numbers, you will be shocking for you. We are 1.4 billion and our total spending on drug and pharmaceuticals is 0.1% of the world expenditure because of low cost, value wise. And what US, Europe and Japan eats, 50%, 50+% of the total drugs produced in the world. That's why every Indian industry wants to sell in US. Because value wise, that is much higher. What you get a strip with worth 10 rupees, 20 rupees, 30 rupees, it is sold at 1,000 rupees over there. And with that earning, Indian companies are able to build up 7-star manufacturing facilities. If a country where ranitidine, which is Asiloc made by Cadila Pharmaceuticals here, ऐसे वहाँ पे लोगों का, which I have been told, they pop up like toffees because due to obese nature of people over there, their living style, now which we have adopted Western system over here, they have huge acidity problem and they pop the pill like this, which now our culture also we are getting into that mode. Now what they wanted, because the 3 regions had a difficulty that we are the best in the world, we are developing new drugs, we are manufacturing new drugs, we are using the best, let us come together to harmonize our regulatory requirements. What are the regulatory requirements? Suppose I have to test the drug on animals. One, USA says test on 10 rats. Europe says test on 20 mice. And Japan says test on 5 guinea pigs. It means industry has to do the 3-time testing to come to the same result. They said let us harmonize these technical requirements so that there is an easy transfer of products from one region to another. And their populations are getting the drugs much faster. And industry has to do 1/3 of the work. So fortunately, the hurdle came in export and import of pharmaceuticals was the differential technical requirements of stability testing, which was taken as a first topic under ICH for harmonization. Because the testing requirements were different and they are long-term studies till the expiry date. So if they are different, there was no way that you have to wait for a new drug which is available in US to comply to European requirements or Japanese requirements. So eventually in 1993, your 1A guideline was brought, which was later revised in 1999 and 2013 as R1, R2. Now try to understand the situation over here. What was happening before 93? This guideline, effort to harmonize, this guideline was taken by ICH in 1990. The birth of ICH was conference, no office. Only people will meet at the conference, discuss, try to develop harmonized guidelines, give job to expert working groups. They will meet at the respective places, somewhere again in the hotels or some places, mutual places, and discuss and develop a draft guideline. No permanent secretariat. That's why it is known as International Conference on Harmonization. Before 1990, it was not tripartite discussions happening. but between Europe and Japan, Europe and US, like 2 regions meetings were happening. Then they finally decided to come and sit together, 3 of them. So virtually they came out with multiple guidelines slowly, and now we had 6 guidelines developed by ICH. These were the person, Dr. T.G. Beaumont, founder of ICH Stability Group Leader, who was assigned to develop these guidelines, Q1A, Q1B, Q1C, Q5C. He retired. He said, I am done with the regulatory science. He was working in GSK Europe, in UK. He said, I am retired now. I am going to Brazil coast and build up a house and spend my life there. I am not knowing the fate of this person. He left the baton to Mr. Nick Turner, his junior in GSK, who was appointed the group leader, and subsequent ICH guidelines Q1AR, Q1IR2, Q1D, Q1E, Q1F were made under his leadership. You should know who are the people involved in this. ICH, we do so much. And I was with them, in the midst of them, in February 2002, the London story I am telling you, that I was on the London street after the conference. So very close relation I got developed with the ICH group, and any issue that my Indian industry, which I could not answer, I will talk and write a mail to Nick Turner. And one of the issues very important was of industry, Indian industry, the question they used to ask, which I asked him, what is happening in GSK, was that I withdraw the sample from the stability chamber. My staff is on leave for 1 week. What should I do with that sample? Where should I store that sample, and how regulatory views that the 1 week window is allowed or not between manufacturing date and storage into the chamber and the withdrawal from the chamber and the analysis. So I wrote to him. He said, it's not part of any guideline, this window system is not part of the guideline, but don't disclose to anybody, I am telling you how it should be done and what is the practice we follow in industry. WHO guidelines. WHO has another set of guidelines looking into the development of 1993 guideline by ICH, which was titled, I can show you the title of it, Stability Testing of New Drug Substances and Products. New, because they were all focused on new drugs and products. In this case, WHO said they had developed a guideline for, as an international now agency, conference, whatever organization they have made. What about conventional drugs and generic drugs which are ruling the rest of the part of the world? So WHO developed this guideline known as Guideline for Stability Testing of Pharmaceutical Products containing well-established drug substances in conventional dosage forms. Not new drugs, but generics. It developed that guideline in 1996. It took so many years that WHO came out with a formal guideline 2009. 96 to 2009, there was no activity in WHO. No emphasis further given. 2009, they come out with stability testing of active pharmaceutical ingredient and finished pharmaceutical products, which further is revised in 2018. Last, you see the same. So 96, 2009 and 2018, it gets further revised. Now this is the guideline of 2009. Totally structure similar to ICH. They say language also should be similar to ICH because all over the world people have started following ICH guideline. We want to extend it to generics. Let us not make a differential, otherwise there will be a huge confusion if we even make a differential language. Make it, but whatever leverage and relaxation we want to provide for generic drugs and formulations, let us insert that. And I was part of this meeting which was held in Cairo in 2008, where we were 10 experts from all over the world to develop this 2009 guideline, which was finalized in WHO headquarters Geneva by the expert committee where I was a member, temporary advisor. Now my whole effort was looking into the fragmentation of Indian industry, how I can help Indian industry while pushing this 2009 guideline. Several elements which I propose were included over here. See how much you can influence the whole industry by sitting as an expert in one committee and help the industry along. And I was not a government official. I went as an independent expert over there, invited expert. Now this is 2018 guideline which was revised, and the relaxation which was introduced because of my presence over there. In 2018 I was not there, they were taken out. And believe me, when I was sitting in the expert committee in WHO headquarters in the expert committee meeting, dominated by Europeans, dominated by Americans, a few, one or two Japanese, China was introduced in my presence when I was an expert member, and one from India. The rest of the world had a very poor representation because these countries were donating huge amount of money to WHO. They had overpowering effect over there. Now, not rest. WHO is meant for rest of the world, but it was that the neck of WHO was because of the donations Trump party did. He immediately announced stoppage of funds to WHO. I am happy that he did that because the clutches of America over WHO goes out. It can think over independently for rest of the world. So now what is happening? An expanded unified set of ICH guidelines are going to come out. It is in the process. Instead of 6 separate guidelines, they are coming out with a single set of guidelines. And believe me, I have told you what was the title of the original ICH guideline. What is the title of this guideline? Original was stability testing of new drug substances and products, Q1AR2. What they are bringing out the guideline, Q1 expert working group, which is now at the, with the expert working group, is stability testing of drug substance and drug products, new they have taken out. Means this is a guideline meant for whole of the world, including new drugs and the generics, same standard of testing. Now whether you are fragmented into small scale, medium scale or a large scale or multinational, same standards apply. No relaxations. So this is the topic which was taken up in 2025. So this is the guideline which came out in April 25 from ICH, which is a modified guideline, new guideline, comprehensive guideline. Now, if you look into the contents of this guideline, elongated contents of 3 pages, because everything has been put into one guideline. And the beautiful part of this is, this guideline also covers Annexer C, Annexers. Annexer 1 is reduced stability protocol design. Thus, Annexer 2 is stability modeling. Stability modeling is a very new concept in the terms of stability testing. What happened in the COVID times? The vaccine was developed and launched within months. There was an expiry date. How to calculate that expiry date when the product itself is new, RNA medicine? How vaccine? How really we do the long-term testing and see its stability? So all through modeling, that you should be able to do prediction and predict the expiry date. They have, I was talking to Mr. Dinesh in flight. He was saying you used a word somewhere when you were in Chandigarh. Can you recall that word? Bayesian modeling. Have you, everybody of you heard of a Bayesian statistical tool? With minimal data, you should have a probability to come to some conclusion. That is Bayesian modeling. World over it is being used these days. But in India, nobody knows where we are. Where we are when something which is being used world over is being introduced into the guideline and nobody knows about it. Then last one, annexer is on stability of advanced therapy medicinal products, ATMPs. Very new technology has come. New technological advancement. You have lot of immunity related diseases coming up. We never had any drugs. Now they are being developed. So these all advanced therapies require new drugs coming up, and we were not covering earlier in the guidelines, which are now being covered. Now, it was on public comments till 2025. The comments have been received already, and you see on the EMA, European Medicines Agency website, the comments are existing, and very interestingly, there are comments from Intas Pharma also. Intas Pharma. They have also put comments. They have read the guideline. They knew that this is going to trouble us. They are into wide variety of manufacture, wide variety of products. And they have put questions to the agency that you really help out and modify the guideline for what query comes to our mind. Now, the important part is this is a schedule, how they are going to when to launch and finalize. November 2026 is the date, step 3 sign off, step 4 adoption and delivery of the step 4 presentation. This year, November, this new guideline gets adopted and implemented, supposed to be implemented worldwide. How many of your company people are following this? That this new requirements with the new format of guideline is coming, and you have to look into this guideline. Whether you are from big company, small company, media scale company, guidelines are guidelines for everybody because we don't have an adopted Indian guideline. Sorry. Chal bhai. Now, there is a website under the name of Stability Hub. There you can see ICH1 draft guideline marks a new era for stability testing, September 2025. April, the draft comes out, is open for comments, and they have started putting over here that what is included, they are starting discussing over here. And one of our pharma professional deep dive, Uday S, deep dive into the ICH-Q1 2025 draft stability testing of drug substance and drug products. So he puts on LinkedIn, deeply looks into this, puts the commentary. How many of us are following this commentary? Tomorrow industry will be in a panic state, I tell you today. If we are not in, we are in August. If we don't start reading this guideline and look into the possible implementation issues, you will be in a difficult position beyond November. Inspections are going to be based on IQ1 guideline, which is getting finalized by November. Now, what are the recommended storage condition ICH and WHO guidelines? Most of you might have seen this table. Okay. Now, what we have done here, we have divided the world into 4 zones. And accordingly, the nature of product, whether it's in a semi-permeable, impermeable container, requiring refrigeration or deep freezer, the guidelines suggest different storage conditions. So they are tabulated collectively. Now, this is ICH draft. I was reading into that critically into the storage conditions. The previous table is from one of our publications. Now, here you will see very interesting that they have given climatic zones. The world is divided into 4 zones. 5th zone also has come now, because 4A, 4B has been divided. There is a long history of stability testing. A lot of politics at the world level has gone into this. 4A and 4B, and a person like me suggested in the WHO meeting, why not to have 4C? Because it talks of 3065 and 3075. And my calculation in my department, with the help of the world experts, what is the long-term stability storage conditions for India? Is 30 degrees 70% RH. We don't have in this table. So what we should do? Either we do testing at 3075, 3065 is not suitable for India because actual condition is 3070. And WHO should list of the storage condition in the WHO list for individual countries. India is, based on my calculations, it is 3070. They have put over there accepted 3070. So I said, why not 4ABC? They said it will complicate further because they don't want to add a single more chamber for India. I have written a commentary about this also, how the politics went over to build up these requirements. But now interesting is see over here, where there is a flaw in this guideline also. It says 30-35% RH. Accelerated conditions 40-75. Okay? 30-65 RH, 40-75. 30-75% RH, 40-75. Now this 30-35% RH is which condition for which product? For semi-permeable container. Now this is semi-permeable container. Water can be in the hot environment, low humidity environment, water can be sucked out. The volume can decrease. The stability for semi-permeable container, PVC container, is supposed to be done under low humidity condition. They are suggesting accelerated under 40-75. But when they come to discuss on semi-permeable containers, they say 30-35% RH same, and right over there 40 degree, not more than 25% RH. But in the previous table, 30-35 RH, they are saying 40-75. Why should not we, India, be the first to comment to the ICH body that you have done this error? Correct your table. My whole emphasis is that we are of sleeping nature. वो हमारी स्क्रूटनी नहीं करते उनकी, या हमें चांस मिलता है, लेट अस शो आवर मसल. हम सोए को पड़े हैं. इंटस फार्मा ने किया है, और भी किसी कंपनी ने इंडिया से अप्रोच किया उनको क्वेश्चन, अपने उनका दिया है कुछ कमेंट? हम फार्मेसी तो वर्ल्ड है, और एक ये नहीं बता सकते कि ये टेबल में सर इसको करेक्ट कर लो. Now, where is the discussion of storage equipment which is we are discussing today? We are on a call of a company which manufactures storage chambers. कहाँ है storage equipment की बात? ICH WHO of 2018 says the stability test equipment must be capable of controlling temperature range of ±2 and relative humidity to ±5% RH. ये condition दी है. And Kesar will boast, sir, हमारा कोई equipment ले लो वो ±2% degree centigrade और 5% RH में आपकी requirement पूरी करेगा. It will do that. Or any other manufacturer will also do that. WHO 218, where in 2009 it was same para which we developed for the first time. So it says something else also. The equipment used should be capable of controlling the storage conditions within the ranges defined in this guideline. ±2%, that is given upstairs. The storage condition should be monitored and recorded. Short-term environmental changes due to opening of doors of the storage facility are accepted as unavoidable. वो आपका audit tail report में automatically comes that door opening, the fluctuations due to door opening. Now comes over here, very important part. The effect of excursions due to equipment failure should be assessed, addressed, and reported if judged to affect stability results. हर word का meaning है. Every word has a meaning. Further, excursion that exceed the defined tolerances for more than 24 hours and their effect should be described in the study report. Now, anybody can tell me, I fought with the whole group, why not 48 hours? I said in India, we have companies which are away from cities, 40 kilometers, 20 kilometers from the cities, and in a very remote area of the country where flights are not there. So is in African country. At least give us 48 hours of leverage that the person, service person should be able to reach the location where the company is located. No. I said we have so much of the big problem of electricity. In Ahmedabad, torrent power is there, rest of the country there is no torrent power. But rest of the regions, US, Europe and Japan, ICH countries, don't face this problem because everything stabilized, power is stabilized at the source. And you have to have put the power conditioning equipment in every facility. Generators and everything. There are no generators elsewhere in the world. Because except hurricane, their electricity is always available. They don't understand India's situation. They insisted, no, we are bound because in that WHO group, more of those people were sitting. I was alone to fight out for the rest of the world. They didn't agree for 48 hours. I said, person has to crack the machine. You are giving 24 hours. It means he has to reach early in the morning when he starts. He will open the machine, find out the fault. Some spare part is needed. Maybe the next day, the next flight, the company sends that part. You can't have everything which is available in the machine and take that as a service person. Again, the 48-hour leverage minimum. Nobody agreed. So, 24 hours remains. Yes. Yes. Mike, let everybody listen to it. Oh, Mike. Come, rush now. अब भागो, बहुत बैठ लिया. अब भागो. हाँ जी.
Came into force, okay, or whatever the issued, then our companies are very good also. Just to follow the guideline, they never notice that, they never put on the paper that our machine went beyond 24 hours. See, if it was not recorded officially, right? If it was recorded officially, and if you put this in front of this WHO with the proper documentation, okay, this was the issue, light went on for more than 24 hours, you know, we were outskirt of the, we don't put those type of things structurally also. So
we
if
are
you
not
see the
truthful.
history,
We are don't have integrity. To
exactly,
tell
that's
the
where I'm
truth.
coming, sir. You know, if you have that data, then you can be arguing even better way, you know, in that meeting.
So question comes over here, they insisted it. And we try to manipulate. We are forced to manipulate.
But
Fine.
why? We don't need to, we need to be honest, and then we have to show this is the problem.
No, no,
and then
you say that. But is another part, inspectors, they are coming for now, the US FD or EMA inspectors are coming regulated to India. Okay. They should have understood the Indian situation. But what they do, they only come to the companies who are exporting product to the US. How about Indian citizenry?
but that's what I'm saying. Even our FDA should make that type of notes and then present to.
They
see,
don't
US
do na. Indian
Yes.
companies are not coming forward out
Yes,
there and
exactly,
express the
exactly.
difficulties.
So, sir, I think we are more.
We are manipulating. अब मैं बताता हूँ. I tell you. First thing I asked Mr. Mohali Patel. First thing. Do you have a patch in your software which brings back 0.5 degree variation to 0.1, which is the limit prescribed, if it is so? What they have done is, some of the company manufacturers have put a patch in the software which normalizes everything and brings closer to. कभी भी deviation नजर ही नहीं आएगी. कर लो. हम भी चोर हैं, बड़े smart चोर हैं. But I'm sorry that this video is being relayed to multiple countries on Facebook. But he said, I don't do it. Now I have to believe his words and salute if he is not doing it. So question comes over here is, अभी आपने उस part को पकड़ा नहीं, which I am trying to say is that you are not captured because it is going abroad. I have to not speak in Hindi. Sorry, I will speak in English. It says excursion that exceed the defined tolerances more than 24 hours and their effect should be described in the study report. How do you look into the effect of that excursion? Do you take out all the samples, analyze them? Then what is left to study? Continue the study for 5 years? If I take all of the samples out, so I ask you, all the industry persons of stability department, stabilitarians, how do we, interesting is that in the ICH draft, this same WHO para in modified form has come to 2025 draft. It says the equipment utilized should be capable of controlling the storage within the ranges defined in this guideline. The actual temperature and humidity when controlled should be monitored during stability storage. Short-term spikes due to opening of doors of storage are accepted as unavoidable. The factor excursion to equipment failure should be addressed, and if reported, just to affect stability results. Excursion to exceed the defined tolerance more than 20 hours should be described in the study report and the effect assessed. Last में लिख दिया इन्होंने. In the previous, it was and their effect should be described in the study report. उसको change करके कर दिया. Describe in the study report and their effect assessed. More emphasis is on the assessment of the effect. Now, how you do, anybody of this hall can tell me how really I assess the effect of the excursion on, beyond 24 hours, on the products that are contained in the stability chamber. Any idea? In this house of 400, 500 people. One or two words in the guideline, and that's why he said pyramidal structure in the industry. Everybody has to, then you start hot words exchange. I interpret like this. Who are you to tell me that this is my interpretation? Oh, Baba, in this interpretation, you will not be able to launch your product for 5 weeks, 10 weeks, because of lack of uniformity in interpretation. Now, if I ask you that ICI 2025 guideline made final in November 2026, how you will affect and assess if the excursion is 24 hours? Yes.
Here is a one point I can also say.
Come here. Come here.
Like all the industry, they are doing these stability studies, and all industry have a different date format. Even in India, people, they are not bother about it. We can go for the single format about the date and timing. We can remain in the all the you can say in India. Somebody is using DD and years in the 4 words. This is a kind of the, you can say, we cannot making a single concerns on the date also.
Now, because he is a person from Kesar and he is going to lot of manufacturers, he has listed lot of problems to me that he faces as a manufacturer with the customers. It is his one. I have intentionally put a few slides on this excursion part. Let us read that. Thank you. Now, this is a summary I have put over here. Last, it is impact assessment for excursion beyond 24 hours. It's a very important. And you are unable to answer to me. First thing is, this person, come here, stand here. Your responsibility is to make sure that every chamber that goes and sold into industry by you, installed by you, never shows excursion beyond 24 hours, that industry is forced to assess the effect of excursion. Yes or no? It is your duty that there should be no excursion happening beyond 24 hours. How you make sure? My friends, buy the chamber from a company which gives you this promise that my chamber will never give excursion beyond 24 hours. But I will say, sir, when I go to the company, they shut the machine to save electricity at 5 o'clock, and in the morning there is no electricity, it goes beyond 24 hours. What should I do? Sit down. I know what is happening in Indian industry because of the fragmentation, small, medium, large scale, and the differential practices. Now, very interesting question came from who? European Federation of Pharmaceutical Industry Association has put a question over here on this paragraph, which was almost lifted from the WHO guideline. It says, the sentence excursion that exceed the defined tolerances more than 24 hours should be described in the study report and their effect assessed, is open for interpretation. Very interesting. It's open for interpretation. It can be interpreted as one single 24-hour long excursion, 24-hour excursion per shelf life, or 24-hour excursion per continuous month. बोलो क्या है इसकी interpretation? It's one time or cumulative excursion in a year or till the shelf life, or it is excursion every month. How many excursions are allowed? Very rational. European Federation of Pharmaceutical Industry Association is speaking this, writing this. Indians itne manufacture, 10,500 manufacturers are silent except Intas Pharma. Anybody from Intas Pharma? Did you write these questions to them? You were or not? No. This is the depth of discussion, first time happening in India, thanks to Kesar. I don't come free of cost here. He pays for my consultancy fee for these 3 hours I spent with you. It's not less amount. It fills my pocket. If I have cash notes, it fills my 2 pockets. ICS does it, brings something, and I get paid to discuss the issue with you. Funny thing. You people are not concerned. I am an academician. Read this yourself. I am giving you the solution. No need to take pictures. These slides are going to be available to you all through PDF, courtesy Mr. Mal Patel. It's okay? Seems reasonable? Okay. Next. If I have stress testing data, I find a product stable if I go even take it to 50 degrees centigrade. If escalation in temperature has gone up to 42, 43, let us say out of 40 degree plus minus 75% RH, fine. I already have a data of my stability of product at 50 degrees centigrade. So I can justify that way. Yes or no? So you should know how to, you have to face the inspector and you have to confidently face the inspector. Any question here? Yes, any question on these 2 slides? Anything you can add from your industry experience on this? Friends at the back, no looking into mobiles. Back benches are always naughty people. Aage aa jao. I'm thankful that you are not talking to each other. The person sitting next to you is the person who will pull you down always. So don't listen to him or don't talk to each other. Are you able to read? Yes or no, madam? Okay. Nah, but I can read it. So small. Okay. Yes. If I put my specs, that also. So, anything you can add on this? I can revise the slide just now. If you feel you are not fully convinced of this. These are solutions which comes to my mind. You got a warning letter?
No, you can get the warning letter, but what you should do in terms of like product recall or anything like that, or what is?
Yes, that is a consequence of it. Either warning letter or product recall. If you are not able to justify that what is this 40 to 45, the temperature has gone, you are not able to justify, they will say simply recall the product. They are wanting to harass you. Only thing is, they are dependent on Indian products. Otherwise, there will be shortage in their country. That's why they give warning letter, correct it, recall this batch, correct it, and then let's move forward. Do you know see the US shortage list. Even potassium chloride injection is in shortage. Such a simple, but it's not that simple product.
Like, can you do the kind of simulation study?
How you do that?
Like you intentionally you put two chambers.
Okay.
In one thing you put you make sure that it doesn't go away. In other one you intentionally.
You want to do crash test with all the products there?
Not all the products, certain products or whatever, just to.
No, no, they are going to talk about the products at movement loaded inside the chamber.
What I'm saying is for the future.
That is what we are calling a stress test. You already have done the stress test. They are supposed to do the stress test.
Stress test, then you have to do it intentionally in terms of like, you know.
No, that you will have chambers and you put the same number and lots of products over there. You are withdrawing more samples, putting in the chamber. Now I ask a question, general question. I will show you in one of my slides. How many tablets are needed to go into a stability chamber for one batch? Anybody? 5, 10, 20, 100? For full scale stability study.
It depends on basically what is the stability specifications and how many products or how many dosage forms are required for individual COA generations of a stability specification. So basically in industry it is generally considered as a 10x as the basically considering reserve samples and other as a 10x, and x is equal to the number of dosage form required for specifications of stability. So that's the general
Give me
way
a rough number, 500, 200, 500, 10,000.
Yeah, sir, generally it will be in 500s or 1,000 dose. So in between of that.
Okay. Practically, Asian guidelines only mentions about the number of tablets that have to go into stability chamber for all full-scale stability study is 1,300. It means 13 strips, 130 strips
130.
of 10 tablets. So that much of the facility you have to create. Now question is 1,300 tablets you are taking out. Suppose, let us say same apply to anti-cancer drug. What is the number that is required? What is the cost of that? How much you can do experimentation by have simulated chambers? Company एक पैसे पे competition पे लड़ती है. And you are saying you add cost 50,000 worth vial anti-cancer drugs. How many you can put in the chamber? You tell me what is the cost that you are going to incur for 3 batches. So we have to give a practical solution that stress testing results. Go for that. Look into those.
So just
Whether they are available in literature or they are available in-house in the company. Hanji.
So just to add, one solutions could be there, because in the regulated market, whenever any product is going to file, there is a requirement of doing a whole time stability. And when the whole time stability data is a compulsory requirement, it may provide scientific justifications for the excursions, because
Whole time means what?
whole time, so during a manufacturing process, by chance, say tablet punching machines is failed, you are storing the entire bulk without a punching, or the punching machine is fine, without a packing, you are holding it. And those hold
Achha,
time,
hold time. You are saying
so
hold
hold,
time.
hold time
But
stability.
hold time will be for what duration?
So hold time stability generally people does for 1 month or so.
So 1 month excursions happened at 6 months in the stability chamber. Will it be enough? Whole time is at normal temperature.
Yeah, that's true.
Not at accelerated or long term at 3075.
That's very true, sir. But some scientific justifications with simulation is may be feasible.
It may be not. Whole
Okay.
time may not be. To define excursion is at higher temperature. You are holding it still at room temperature.
Yeah, when sir 40°, 4075 is there, and the excursion, say by chance the chamber is down, the temperature of a room is lesser than 40°.
Excursion at low temperatures is acceptable.
That's
It
I'm trying
is
to say.
adverse condition when it goes towards the adverse side. Then it is an issue. Real sense that effect on the product. Lower temperature, you are already doing 3070 study,
Correct.
3075 study. So 4075 chamber fails, comes down to 3078, nobody will ask the question.
Okay, yeah, got the point.
So it has to be scientific and rational. Now, essentially a stability chamber is not guided by stability guidelines truly. It's a CGMP mandate. So what it is: design, installation, operation, calibration, performance, qualification and documentation, maintenance of stability chambers, revalidation and cleaning protocols. Now somebody asked me, I was giving a lecture on stability study somewhere, and one of my regulatory friend, Dr. Kapil Bhargav, late Kapil Bhargav, asked me a very simple question. Sanjay, can you explain at what frequency a walk-in chamber should be cleaned? A very interesting question, which I had no answer, immediate answer, because industry practices vary a lot. So there is no guideline on this. And you will not find in CGMP any guideline on a specific requirement of a walk-in stability chamber. Answer on this. It is SOP driven, guided by quality assurance, which should be here to understand what has to be done. Now, if there is a spillage, see, it is a controlled environment chamber, very controlled environment chamber. Temperature, no dust should come until unless it is put where AHU has been put. Stability chambers are installed on the floor where AHU is there and all dust collection is there. That cleaning is rapidly to be done. But if it is put in an air conditioned and AHU environment, stability chamber where it is getting installed, that will define how much dust collection is there and what kind of products you are handling there. If you have taken a box somehow, the box base has got weakened, the bottle has got on the floor and broken, and there is spillage of syrup, you need cleaning immediately. You can't keep it and come the whole thing fluid coming under your foot, under your shoes. So these are the nature of that you have to define internally. And other thing is very interesting, how at what frequency you open the stability chamber and walk in room. If the dust somehow, because outside, if it is a clean environment, pharmaceutical companies don't allow even a speck of dust to come inside. If that is environment, no issues. But if you are opening the environment, the chamber opening to the environment which is dusty outside, and the frequency of opening the chamber will decide how much dust is going inside. So all these things you have to see the installation where you are going to do it. Ask him, sir, come here and tell the worst type of installation you have done with your hands. Batao jara, audience ko. You are a stability chamber installation person.
Even people, they are talking about the perfect IQ, OQ, PQ of the documentation, complete documentation. And most
loud,
of the
loud.
And most of the cases, they are not providing the proper environment, water quality, and continuous electricity. But they required all kind of the, you can say, validation activities completely fulfilled by ICS guidelines within the limits. So worst condition is the water quality is a major part of the instrument which is required, electricity, continuous electricity, and we can say the environment. Most of the cases we have seen the people, they are not controlling the environment. Like we have already said that AC, they close in the evening and they start in the morning. So kind of the things they are providing.
So he was showing me some pictures, raw floor. And they want that instrument should be, the chamber should be installed, walk-in will be installed over there on the raw floor. And one of the best companies in India. I don't know. If you sit with him, I had spent a 1.5 hour flight from Chandigarh coming down to Ahmedabad with him. Horrible stories where people want the stability chambers to be installed. So, I don't know. There are a lot of factors where our industry person like technical person like you have to learn a lot, and you talk to your management, sir, don't be a lalaji, be a technocrat if you want to make formulations. It's a very flat I am taking up. लाला जी को 50,000 रुपया रोज चाहिए पॉकेट में, जैसे मर्जी लाओ, उन्होंने मैंडेट दे दिया. Pharma doesn't work like this under regulated environment. So you have to spend money to earn something under pharma domain. Now, disaster recovery plans, I will be, principles of monitoring frequency and documentation, disaster recovery plans, I will talk later in detail on this. Now, specific recommendation for photo stability testing in the guidelines. Let us come to Q1B. So, the highlights of the guideline are that you should have option 1 or option 2 equipment, ensuring recommended exposures, light intensity and wavelength qualification, sample presentation, result interpretation, documentation presentation. This was the most beautiful thing ICH did to harmonize the requirements of photostability testing. I have seen people exposing under UV, where we sterile the microbiological plates and we put UV lamps. I have seen people putting those UV lamps, putting bulbs which give heat. So primary, when I was there as a student, variety of these type of situations I could see was photostability testing. No control on exposure time, no control on the type of lamps. Beautiful thing they did there, at least gave a standardized protocol for the industry. So 2 light options. Option 1, artificial daylight xenon or halide lamp with filter for radiation below 320. The reason is our ozone layer cuts the radiations below 320 nanometer, 330 nanometer. These layers below this are cancerous in nature, and so we are protected due to this. Option 2 is combination of cool white fluorescent and near UV lamp, and where this spectral distribution is defined in those lamps. Now, the inspector can come. Do you have spectrograph in your company to prove to me that your spectral wavelength is between 220 and 320 and 400 nanometer? That your lamp that you have installed is between giving this wavelength? And 99% Indian industry doesn't have a spectrograph. Then maximum energy emission between 350 and 370. Minimal light exposure, visible 1.2 million lux hours and UV 200 watt hour per meter square. So question is, first thing I bought when I bought the photostability chamber, the revised one from Binder, I said I want lux meter and the UV meter. So do we have those? And calibration of those. Thanks to Mehul, that Mr. Mehul has a perfect NABL lab to test all those, whatever is required for testing visible and UV light. In ICH draft, interestingly, they have put the same requirements as the old guideline, but they have put the option 3 over here, which says ambient mild light conditions, predominantly light greater than 400 nanometer during manufacturing, processing and in use for which a fluorescent or LED lamp is recommended. Now this question comes very oftenly to me. Can we replace the previous option 1 and option 2 lamps with LED lamps? Because everywhere we see now the LED lamps. Why we are testing still the tube lights, UV tube light or visible tube light, daylight lamp? First time they have added that LED can be used, but not in the chamber. Light chamber still have to do the same. These are because in the manufacturing area, you are using LED lamps now. Some sort of confirmatory testing can be done by using the LED lamps. That yes, the product is light sensitive or not. So option 1 equipment is on the left side, option 2 equipment is on the right side. Option 2, photostability chamber with control of humidity and temperature and humidity is displayed over here. Multiple companies manufacture this. The chamber photostability we had in our lab also was of similar kind, with temperature control, humidity control. Now, what we observed, we bought these stability chambers and photostability chambers not as a showpiece, because we were not in the regulated environment in the academic institution. Our purpose was very different. We were doing research. So my question came when I was talking of India specific. My question in my mind was, why stability chambers are dark from inside? Have you ever thought of this? That whatever Mr. Mohali makes, other than photostability chamber, walk-in rooms are dark from inside. Is our environment outside dark? Funny. I said, let me do one experiment. I took a hygroscopic drug, ethambutol hydrochloride, put in a dark chamber with, let us say, 40 degrees 75% RH. I took a photostability chamber set at 40 degrees 75% RH. Same light exposure, 1.2 million lux hours, 200 watt hour per meter square. Bring out the samples and let us see what happens. So, you could see that difference in gain of moisture by drugs and excipients in light as compared to dark. In ethambutol hydrochloride, almost 28% higher moisture was taken by ethambutol hydrochloride in the presence of light. First time in the world we reported a physical phenomena happening under the light. Always we talk of photolysis. Light effect on degradation of drug. Never anybody has thought that it can lead to increase in the rate of gain of moisture. And when my paper was published, I found that citation coming from a very different place. In the semiconductor industry, when the chip making industry, they see when they are doing chip manufacturing or your semiconductor industry, there is certain light in the room. And they are finding small droplets of water over there on the chip, where it should not be. It should be totally under dry environment. They have quoted our paper. Can you see the magnitude of our research impact? Just observation between dark and lighted chamber. I asked the European people in WHO, why Q1A and Q1B? Why not our chambers are lighted from inside? They say, you are creating another mess for us. For 4 long years of study of continuous chamber running, when we put light in a walk-in chamber or chamber like this, standalone chamber, the influence of the light would be that there is a decay in the lamp intensity. You are creating another trauma for us, whole industry, that now another calibration and qualification of the light intensity, which is decreasing all the time. So we kept, if the light is sensitive, you do the protection, Q1B was separated out. And they said, we don't have the light enough to. The other thing was, they said, I don't know about the practices in your country, good or bad. In our world, in a chemist shop, pharmacy, where it is written outside pharmacy, not a chemist shop, only on the display are your OTC products. Deep inside, a pharmacist in white coat is standing, sitting. And all our products are in secondary pack. In the drawers, there is no light falling on the products. And that I had to shut my mouth. What I should, they have a good practice of retail of drugs.
pharmacy only, you know.
Sir, I have been to multiple airports in the world.
I practice as a pharmacist in US, and I know that
So
it's a wrong thing.
How it was? How it was?
It is open in the bottle. They put it in the open only, you know, like in the in the light only.
Nothing. When they hand over to patient, it is in a bottle.
It's in the amber color bottle they usually prescribe. In the amber color bottle.
Yes.
Yes.
But original product is in a multiple bottle.
Yes.
From where they take out certain quantities.
In the big bottle, okay, it's open in the light, and you just count it.
Sir, that is hospital pharmacy.
No, even in the retail pharmacy.
In the chemist shop,
in the retail pharmacy. I work as a pharmacist in the US for few years.
Okay. So do you feel it's a good practice?
Means if you say this one, yes, then it's not a good practice, you know. If you
our shops, if it is facing east side, when the sun rises over there, direct light of sun is falling on our products. It is not happening there.
Yeah.
It's not happening there. So there is a hell of a difference between a tropical world and
but as you said, like, you know, there is artificial light. There is artificial light also over there. They said it's not that. I'm just arguing on based on that.
Artificial light is mild light, very mild light.
So this one done on the artificial light only.
That is our. I will show you. Just let me finish, then we come back to this discussion. So ethambutol. Ethambutol hydrochloride, when I put in a chamber, you see this is without light, the lower one. The total moisture gained by ethambutol hydrochloride remains the same after a certain time period. But the rate of gain of moisture, you see how much it has gone up. This was naked ethambutol hydrochloride in a beaker. And this is also showing the combination of 4 drugs. Ethambutol alone with isoniazid, rifampicin, isoniazid, ethambutol, we work on anti-TB drugs. So we took this combination and 4 drugs, pyrazanamide included. All of them are showing increased rate of pickup of the moisture. Wherever ethambutol, and ethambutol, what it does, it's a ethambutol hydrochloride. It is hydrochloride component is corrosive when in aqueous environment. It is corrosive to the foil, aluminum foil. It creates pores in the aluminum foil and takes up the moisture from the environment. And our Indian pharmacopoeia doesn't declare it as a hygroscopic drug, mind it. Very funny. Now this is exposure to 4075 anti-TB formulation taken naked from the market, and 5 days the spots appear due to ethambutol hydrochloride taken moisture, naked one, in dark. When I put in a light chamber, within 5 days, see the bleeding what is happening of rifampicin dissolved in water taken by ethambutol hydrochloride. All papers published in International Journal of Tuberculosis and Lung Diseases. And lower one, a tablet, another brand tablet, cracks in 5 days in a normal chamber, dark chamber, and see the lighted chamber, how much it gets worse. Now you say, sir, primary, the direct product, if you put always, it is going to show some problem. Come to the packaged product. You can see over here, funnily, a droplets comes under the light over here. See over here, even in the packaged product, when exposed to light. World has not taken the light seriously that much, because it is not the means real interest issue with the ICH countries. And see, this is Pfizer product. When it is in control, is the top one. This is Boehringer Ingelheim product. All international companies. When I put in the dark, there is some weathering of the, and the packaging is light, the print color is going off. But when put under the light, see the weathering that has happened, and the product which was branded Jericor, and this, what they have put over here is all faded. It means they have not qualified the red ink on the package under the light. same amount of the hours which a dose of ICH dose of light in the
What I'm
chamber.
saying, how many days of like of light exposure?
Days required, maybe 5 or 10 days, whatever the dose in our chamber, 8,000, 9,000 lux at one time, and the hours that were needed for 1.2 billion lux hours.
It was just 8 days.
Until unless we do intensive studies, we will not come to know what is happening. Now, what has happened is the start of the topicalization of ICH regions. See, where is the ICH region? Here on the top, this is ICH. And this is rest of the world. So, rest of the world is lighted, and ICH regions are having very low light. Mehul said, I have visited almost how many countries he has visited. He said, I don't see much light when I go to the ICH regions. And it's a celebration. Even it happened with me. I have been to Europe, I have been to the WHO multiple times. If sunny days there, it's a celebration. But now things are changing. There's a sunlight directly overhead is equator line. That's why African countries, coastal countries, they ask for 75% RH in 30 degree. Low angle of incoming sunlight, as we come down from North Pole, comes towards the equator, the light intensity increases because Earth is closer to the sun. इसकी बैटरी डाउन हो गई है. Now, what is the changing dynamics? Heat wave scorches western states of US. Triple digit temperature forecast to set records 0007. Means 2007. This was the news. And see the people, this girl on the right side. What is the fate of, what is happening? Protecting herself. They are not used to such heat in US. Death toll rises in southern Europe heat wave, again dated 2007. Here, 2007, 25th July 2007. Doesn't die in Japan heat wave, July 27, 2010. Now come 26. The 26 Southwest US heat wave was one of the 6 most astonishing weather events of the century. Western Europe has hottest June on record, 9th July 2026. Japan expects even more extreme heat August 1 of this month. ICH now is facing tropicalization, ICH regions. I think in very few number of years they are going to understand that what SARANJIT said at that time is going to prove true. The paper by whom? United. They already started thinking on those lines. Heat profiling, heat proofing, health products adapting to a hotter planet. The growing risk of climate driven heat and humidity challenges. Chapter number 4 of this document. Now they will realize what SARANJIT came from tropical world and told us, gave us a warning, why your chambers are dark in color, and it's a very important issue. Nobody talks about it. Nobody realizes the importance of this. Effect of increasingly hot climates is intense light always accompanies intense heat in the environment. बाहर 50 डिग्री सेंटीग्रेड है, लाइट भी उतनी है. आप अम्ब्रेला से लेके लेडीज को चलते ऑन द रोड, यू विल सी द लेडीज ऑन द रोड हैविंग अम्ब्रेला इन हैंड. It's not only heat, because to protect the light. Yes or no, madam? You use umbrellas when it is very hot outside. Now stability chambers are used by car automation industry, car companies. Earlier Tata, Lord Indigo, whatever is the brand Tata, when it brought the first car, in 10 years or 5 years, you might have seen the weathering of its paint. Now today paint industry, by using the stability chambers, have come out with light chambers with a paint that doesn't fade, doesn't get bad. It's a reality of life. What is the difference between the light intensity in our country, zone 3 and 4, versus the light intensity in Europe? Whole year, light intensity 50,000 lux on any summer day in tropical world versus 1 week of light in an Asian country is equal to total light in a year in a country in Europe. How we can? They were not worried. They kept light out of ICHQ1, created Q1b. They didn't realize this. They were not interested for you. They were interested for the 3 regions. Now that their guideline is not labeled as a new drugs and products, ICH draft 2025, they should have realized this, but they are adamant still. Because the chamber's light intensity goes down. They don't want to get into that. So, a justified progression. Again, I am saying something novel over here in this hall. Propping you as Indians, think out of the box. So, in temperature Arrhenius testing, initial years of scientific expiry dating was based on Arrhenius formula. Do temperature studies, extrapolate to 25 room temperature. Study at 50 degrees, 60 degrees, 70, extrapolate down to normal room temperature. Then came the concept, somebody thought, Joel Davis in US FDA, thought, yeah, environment is composed of temperature plus humidity. So why we are keeping humidity out? Earlier there were only ovens, but now we have humidity chambers since that. Joel Davis in 1972 thought of that. I have original papers of US FDA where Joel Davis proposes 4075. Now the time is temperature plus humidity applies, which we call as a global stability testing. Now they agree, if you have done the study at 30 degrees 75% RH, you don't need to do the study at 2560. Because that is more adverse than 2560. You see the whole guideline, it says, if you have done the studies, that is global stability testing. Now with this hottening of the world, this is the right time that our chambers are light, having light, at least normal light for long-term studies. Otherwise, how do we improve the light uniformity in a chamber? If somebody has tested, Dr. Mehul, Mr. Mehul, you are a doctor in your engineering field. You should have been given multiple honoris degrees. How much of your chamber shelf gives you light of same intensity? It will be only in the center, a small pocket in the center. So how do you increase the width? Because I have lot of products exposed to photostability. So either you have a light bank under each shelf. Okay, that is one. Second thing is to increase what international companies are doing, that they have filters placed over here or this filter. So it suppresses the light in the center from a very small area of light exposure, high intensity light exposure, it widens using the filter. decreases the intensity of light little slower. It's a just perforated sheet for UV and visible, separate type of sheets they are using. So follow up this, if possible not being done, try using it and do it and create a chamber of that kind. So coming to what is the manufacturer, I am happy you are here. Now don't look to this side, look to that side. You will better read directly from that. This angle is a longer angle. Okay. The company has to do development of stability chamber specification documents such as design, functional and user requirement specifications, develop and execute of high quality stability chamber validation protocols such as installation IQ, OQ. Now I have put over here, you normally say IQ, OQ, PQ. Nobody puts CQ in that, because all your documents have to show, which you do in NABL laboratory, that they are duly calibrated. Traceability of your calibration has to go along. Okay, that I call as a CQ, calibration qualification. That also documentation chain has to be there. Whole traceability you have to provide. Then comprehensive validation report package, summary of all test results including deviations, the executive protocol, test data, the test equipment calibration data with NIST traceable calibration certificates, the validation report package prepared and reviewed by chamber manufacturer is submitted to client for approval. So this kind of, that is their responsibility. Now what are the specification documents required? Basically design specification, functional specification and the user requirement, URS. Okay, what you suggest and what you want from the chamber and what kind of chamber you want, what are specifications for those chambers. Now what are the responsibility of the users? Very, very important. Their responsibility, you ask them. When Mr. Dinesh Gupta goes to the company, company asks them 100 things. But users don't take any responsibility. It is you install, I will have paid you money, it is your 100% responsibility. I have heard from different manufacturers, same complaint. Very interesting other demand comes from the industry. Every person will say, sir, chamber mapping, do it for 1 month because you have to run the chamber for lifelong, of life of 10 years, 15 years. Do the mapping with 20 sensors for 1 month, 2 months or very variable time periods. And they are really worried about it that every company, there is no standard protocol, everybody is asking for different mapping SOP of theirs. They came to me, sir, can you discuss with the audience sometime, not this time, previous, one of the lectures which was sponsored by the company. They asked me, sir, can you sensitize to the audience requirements of the mapping of the chamber? How many sensors, how much time is actually needed for a validation of mapping of the chamber in the set conditions? I asked the audience, you are buying a new chamber. They are giving you all the documents, fine. Still at your location, yes, they have to do for walk-in chamber, they will do the mapping because they bring in the small parts, they assemble over there. They have to do the qualification mapping at the site. But small chamber, they have tested, qualified everything in their facility. Yesterday I saw how they are doing the qualification, mapping through multiple sensors. Question is, I ask you all, and challenge me if I am wrong. Who has bought car in last 1 month? Let me know. Anybody of you has bought car in last 1 month? Or last 6 months? Or last 1 year? Raise hand. Please raise hand and stand up. Raise hand and stand up. Or 1 year or 2 years, 3 years. Anybody has bought a car in 3 years? Please, sir, you bought it? Okay, stand up. My friend, good friend, sir, we are of equal age. Come here. Sir, congratulations on that, buying that car. What was that car model? High Rider. Wow, 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 wow. Congratulations, you could afford that. I could not. Now tell me, did you went to the dealership and tell them, sir, I want to test this car on road for 1 month before I buy it? एक राइड लिया वहीं पे 5-10 किलोमीटर करके वापस आ गए. That was the only testing you did. In that car, your most loved ones and you as most loved one for your family had to drive that car. Yes or no? It's a most risky device, transport device, in which your family sits. Why didn't you insist the dealer, I want to test the product for 1 month on road before I buy it? You tell me. Qualify for 1 month on road before my eyes for this specific car before I buy it. Did you do that, all of you, when you bought a car in which your family rides? Yes or no? Think over for a while. And you as a pharma company ask them to test, validate for 1 month with 20 sensors inside before we really accept this chamber. Is it logical or not? Think over. You believe in the company, na? Believe in Mohali also. For the good job they are doing. Go to their company, visit their company before buying the chamber. Look into the practices they have. Believe in them. बाकी है तो है ना हमारे पास. जब रॉन्ग होगा तो मार लो, पकड़ के मारो, नीचे मारो, पीछे मारो, इधर मारो, जहाँ मर्जी मारो. मेहुल बहुत स्ट्रॉन्ग है इस मामले में. सॉरी यार, आपकी स्ट्रेंथ देख रहे हैं कितना मार खा सकते हो. How much you can sustain the beating in English for your friends overseas? Thank you, ji. So nice. I am sure you are enjoying the car. High rider is a high rider. High risk rider. It's not a high risk rider. So we have to be logical. Sometimes in the interest of our things, we don't understand that what will magical happen if we are accepting the validated, pre-validated chamber and start working on it, using it. Why insist the manufacturer for 1 month of mapping with 20 sensors, unnecessarily increasing the load, they have put manpower. They are into, Mehul ji, idhar aao, aap bhi aa jao. Aa jao, please. Congratulations for holding this session with so many people in the hall. Now you tell me, sir, ऐसे ऐसे अभी दूसरा पैर नहीं आया. Now you tell me how much you feel a pinch while facing competition with your other manufacturers of saving every rupee that you have to focus on financial management within the costing of your chamber because you are in a big competition, and you say with your word, extra mapping hours is a load on me, yes or no? Yes, thank you. Have a nice day. Sir, आपके सामने मैंने करवा दिया manufacturer को. We have to be rational. I am giving a logic. If you are having your bestest family member sitting in that car and you don't test on the road for 20 days or 1 month, because it also has to last for 15 years with you, like the life of chamber. I am running a car which is 213 year made. I cannot afford high rider, sir. Sorry. I am a petty, poor father of a rich son who runs a startup, who has a startup company worth 1,000 crores. But I am a poor. Only thing is brings me Brook Brothers shirts for me to go to such events. I'm a poor father because I earn only 1 lakh pension. And help they provide me, financial help they provide me. My all cars till date are old second hand. What? सर अगली बारी मैं इनके ऊपर डिमांड डाल दूंगा, मुझे कार दे दो गिफ्ट में, तब आऊंगा। आप इनका खर्चा बढ़ा रहे हो, मेरा नहीं बढ़ा रहे। आप दुआ करो ना भगवान और दे आपको। नहीं देखो ऐसा है, पहले मैंने कार ली सेकंड हैंड, टीचर था, सैलरी लो थी, बच्चे बड़े हो रहे थे, सेकंड हैंड गाड़ी ली। फिर मेरा कुलीग एक जा रहा था न्यूजीलैंड, वो कहता है यार मेरे ब्रदर को मैंने किया था, कमिटमेंट की थी, दिल्ली में रजिस्ट्रेशन नहीं हो रही, अब मैं कार कहाँ छोड़ के जाऊँ, मैं कहा मुझे दे दे, वो कार आ गई। फिर बेटा एक गाड़ी नई लेके पढ़ने चला गया था बॉम्बे, नौकरी लग गई उसकी, ही वॉज वर्किंग इन मुंबई, उसका था मुझे कार चाहिए, उसने कार ली, मोहाली से ली, ले गया बॉम्बे। दैट ही रिटर्न बिफोर ही वेंट टू फ्रांस फॉर अनदर MBA, वो हो गया। नाउ ही हैज़ अ कंपनी कार, he got his Mercedes, उसने मुझे his Kia Seltos, 3-year-old he sent to me. So I am unlucky person. I have to have a second used car all the time. And today, that same car, Kia Seltos, after 6 years, it is in a service center for the noise in, thanks to ethanol 20. So, now, read into these, please, for your own interest, what are your responsibilities? Are you in stable state, all of you? All of you are stable? Okay, enjoying. Oh, good. Medicine तो ये भी खाते हैं यार. Now, these people, I tell you, really face the problems with the user responsibilities. They don't give supply of water, proper water, location of installation of the chamber. Engineers are not fully trained to handle the chambers and solve the problems. Yesterday at the airport, he's getting call after call, or call after call. And he was providing one simple solution. Yaar, put your computer on again. Switch off and then put the computer on. Why can't they do on their own this? Yes, it was the same thing happening. Now, we are already 7:45. I have to finish in next 30-40 minutes. Now, type and number of chambers. How many are needed? What kind? That's a very big decision one has to take, depending on the level of the company, whether small, big, or large, or whatever. So, depends on the variety of storage condition that you have to choose what type of products you are manufacturing. Whether you are manufacturing any product semi-permeable container, then you have to have low humidity chambers. But otherwise, long-term accelerated intermediate testing you need essentially. 25-40, 25-60, 30-25, 30-35, 30-65, 30-75, 40-75 cabinets of high temperature, storage at 2 to 8 degree, -20, -40, -80, photo stability, specialized conditions. What example I put? Today, I have entered this at 3 p.m. into my slide because you have a professor here in Nirma University. She just went to NASA and came back because she is working on a research problem. Yeah, she has taken a research problem to look into stability of pharmaceuticals which accompany astronauts into the spacecraft ISIS. What happens when the radiations of a very different kind fall on pharmaceutical products? They penetrate even the very thick space ICS, the spacecrafts, and the what we call as the space station they have built up into the space. So, like testing of stability of pharmaceutical in manned spacecraft. Now, type and number of chamber required depends on the scale of operations, including decision on standby systems, redundancy we call. Added set of chambers, ovens, water baths are a requirement of sample stressing, accelerating aging, transport stability, and in-use stability. Now, this slide also I have added as an example to you how a stability testing misadventure comes to a situation that company is lost. In 2007, on 23rd November, Ranbaxy had a voluntary withdrawal, a first for an Indian drug company in the US. 73 millions of gabapentin tablets. How many are 73 million tablets? 7.3 crore tablets were asked to be recalled from US market. ऐसे नहीं कंपनी बिकती, the companies are sold due to these shocks. Ranbaxy said found related substance in the tablets that were beyond the limits. Because impurity level has gone down, there is a lactam impurity degradation product that is produced and it had gone up than the limit they have agreed upon. Now what happened? Inner story, the big concern was it was a class 3 voluntary recall for 7.5 crore tablets, and these are the class 3 recall means products that are unlikely to cause any adverse health reaction but violate FDA labeling or manufacturing regulations. No concern of health to patients, absolutely. Still, they were asked to withdraw 7.3 crore tablets from the US market. Now, see the cost of manufacture, cost of transport, shipping, it went from India, transfer, the cost of recall in US and cost of disposal. It's a potent drug, where to dispose? You put in sea, bad for fishes and human beings that swim in the sea. If you put on the landfill, it recycles after rains comes to your water chain until it decays. Bad for human health. Where to dispose? If you burn it, it goes into air. All chemicals go into air. So, what was the issue? This is an era when impurity guidelines were developed. ICHQ3 guidelines were developed. Issue was, original gabapentin innovator was Pfizer. Pfizer was allowed 0.5% beta-lactam impurity. When Ranbaxy went as a generic player to US FDA, they said for innovator we have already has a limit of 0.5%. We will not give you because you have to ship the product from India to US. For safety margin, we will give you only allowance of 2.4%. Okay. Now this person, Ranbaxy person, asked his stability team back in Gurgaon, what is the level going on in the product? And by that time, the product has undergone 1 month stability already, 1 year stability already. Maybe till they had approval process going on for US FDA hobnobbing and this, they had to bargain the impurity. So this US FDA said, we will allow only 0.4%. He asked his technical team, what is the level of impurity going on and what do you expect at the end of expiry? They said it is around 0.23%. You safely take 4.4%. And when actual value and stability study, when they were completed, the product went to 0.46%. There was small misjudgment how much of the trending that what will be the impurity value at the end of the expiry date. So when it happened, there was no, because already approved product was 0.5% by Pfizer. They give leverage to them, not to Ranbaxy. The bargaining had to be done by Ranbaxy with the US FDA to come down to 0.4%. their impurity went to 0.5% at the experiment. Class 3 recall with no effect on humans. Just because you have agreed with US FDA on stamp paper, you have to withdraw 7.3 crore tablets. I asked my friend, my class fellow who was working at Ranbaxy quality assurance at that time. He told me this story, inside story. He said, don't worry, Saranjit, there is no commercial impact. Although US FDA was badly taking it Ranbaxy internal practices, that eventually Ranbaxy had to be sold to Daiichi. There were instances after instances, more on stability reasons. There was another product in which they found that whole of the data was, sample was lying untested in Pontasa facilities. They have withdrawn from the sample, lying untested, but the whole data of stability data generated on computer has gone to FDA. That was the internal malpractices. Beautiful company, which was a training ground for all the professionals in this country, which has raised all the companies, but integrity somewhere in the staffing brought this company down. So question comes over here, could be the situation provided a better approach? Could I predict? I could have said the team sitting in US FDA, wait for 2 days, I will give you the what exact value is going to reach. Don't wait for long-term studies. Can I give within 2 days? This is modeling that has come in the new ICH guideline. Can we rely on some modeling type to really assess what will be the exact level of impurity because you were bargaining over there? So what has happened is, Dr. Waterman, who was working in Pfizer, after he was pushed out of Pfizer because of they had thrown many people out of Pfizer at that moment, which were less wanted. So he's a physical chemist. I met him in US. So he has brought very interesting Arrhenius plot equation in which he has added log K is equal to log A frequency factor minus E is activation energy divided by RT value. He add humidity factor into this. Believe me, this is Arrhenius equation. And he has added a constant with percent RH. And I talked to Dr. Waterman, why don't you add a light over here, plus light factor? He said, I will think over it. So he says, and I tell you, Arrhenius equation, Arrhenius got Nobel Prize for it. I feel Waterman is one person who should get lot of prizes for this. So 3 parameters need to be determined using multilinear regression for each degradation reaction. So here what he has done, conditions and duration SF program protocol, which is called Accelerated Stability Assessment Program. In his protocol, what he does is, why I am talking in this whole stability seminar, testing for storage facility, he says API stability protocol, he has to test for 0.5% degradation. So he says temperature and RS 70 degree 5%. He did multiple experiments. Now he has combined temperature with humidity. 70, 75, 85, 80, 40. He found that 0.5% impurities reached in API in 14 days. When it goes to drug product, 50, 75, 14 days, 60, 40, 14 days, 75, 14 days, 70, 75, 1 day, 80, 40, 2 days. Within 1 day, you can predict whether your product is going to have that lactam impurity after 2 years, if it will have 0.5% or not within 1 day. This is the element that has been added in the modeling section. They talk of accelerated stability program, use of accelerated, not assessment program, but accelerated stability program in the modeling section in the 225 draft guideline. And this is the program which whole of the industry in the Western world, the innovator industry is using this program, and maybe it's 1 crore to Indian companies, software that. Had Ranbaxy at that time had some model developed in-house to predict 0.5% degradation. How long it will happen? Within 1 day. Wait, be there in the hotel, will come with a more valid result. Then just imagining it is 0.26% will reach 0.4, okay, go ahead, give and bargain over there. And the company, you see what Sun Pharma has bought finally, this company. Any questions? So, question is, how do we test now? For doing this testing, you need some device, some storage facility. Mr. Mehul says, okay, buy 3 lakh worth of chamber, multiple chambers, buy it, temperature, humidity, different variations you want to do, run the experiments, buy my chambers, I am very happy. But one company has brought this device. You have ovens for other kind of studies, simply ideal for ASAP, Accelerated Stability Assessment Program. This is a device in which you have ovens already placed, 50, 60, 70, wherever you want to do, place the samples. Take the tablets in one, it is a bottle with a compartment, 2 compartments in which with a salt solution to create certain humidity, and in other you put your tablets. And this top cover is a wireless sensor technology. So put these small bottles with different humidities in the 60-degree chamber or 80-degree chamber. You can monitor the set condition inside the bottle on your laptop software, monitor what are the conditions going on, and based on take the sample out tablet and test it after different periods. You will come to know whether 2 days or 1 day. So complementary storage facility has been created. This bottle is not still available in India, wireless system. So you have to import it. Yes, anyone? Yes.
It can be approved or just like it's for your internal study only?
If your modeling allows this, and you can convince your inspectors that we are using, this is a company known as Amebis, and we are buying this wireless system with whole the record coming on our laptop. Okay, there is no reason that US FDA inspectors, because this is not kind of regulatory study.
Okay.
This is for internal
okay,
assessment. See, I have to test 4 formulations. Which formulation gave me 0.4% instead of 0.5%, 0.2% instead of 0.5%? How do I check?
It's not mainstream right now for the regulatory purpose, but it's for internal thing. Okay.
Now, optimal display, monitoring and recording devices. Normally chart recorder were there. Now we have lot of data logger systems, computer, separate data loggers and display systems. So they have certain now all data coming in their PLC or whatever it is, display systems. What we call as a controller? PLC controller? PLC, yes. Then you should go for precision of storage tolerances. Now to get plus minus 2% in a walk-in chamber of 10,000 liters, you need the controller. Now this is technical specification theirs, which says temperature range 20 to 60, humidity range 40 to 95, accuracy is 0.5 degrees centigrade, plus minus 2% RH. Now it is 2% of 0.5 degrees centigrade, not 2% RH of the chamber. So if you had to take 2 degrees centigrade plus minus in a walk-in chamber of 10,000 liters, your controller should work at 0.5 degree, control at this level. Uniformity plus minus 20 degrees centigrade and 3% RH. Look into other vendor. He gives the temperature control at 0.2 degrees centigrade. And control resolution 0 degree and temperature uniformity shows a 0.7 degree centigrade. Humidity humidity control is at 0.3% to get even a 3% plus minus 5% RH, but for better accuracy, because earlier physics didn't allow humidity controllers less than 3%. That's why 5% limit was there. I think that also is going to be tightened in few years, because we have now Japanese have developed controllers which can control humidity at plus minus 0.3% RH. So these controllers are available in the market now freely. So now we can have a better humidity control in the system. So this, what they offer or any other manufacturer is offering, you have to compare who is better specification wise. Then security and alarm systems. There are a lot of multi-levels of alarm protection that you can get. Just read it through quickly, have an idea of what, or I will, you can get the PDF of my slides later and you can read through what are all the alarm systems. There can be glow of lamp, there can be a beep, ringing of bell, auto dialing, flash of message on PCs of all operators. In between, it will override all the operations that are on a PC monitor and give you a flash that it is going out of range. Then record and documentation of event. Delays are needed to be provided in alarms to avoid unnecessary notification. Door opening condition is not registered unless the door is open for more than 5 minutes. So continuous door more than 5 minutes, it will record the event in the audit trail. In case of fluctuation, the system allows say around 20 minutes for system to re-equilibrate before setting an alarm. Now if you open the door, let us say 5 minutes is the limit, you take 3 minutes, there is some disturbance in the chamber. How quickly it recovers the set conditions? That is the efficiency in the system that is built in. So this is a very critical factor before you purchase the equipment. What is the recovery time? So the alarm might be cleared only when a user ID and password is entered. Not anybody can clear the alarm. So calibration equipment options, temperature, humidity, lux meter, UVA meter, spectrograph, and another thing which I have added into is anemometer. Anemometer is one device which captures air velocity at a very low velocity also. It has a fan blade which is very light. Just small airflow, it will capture the airflow and give you what is the air intensity. Now what is the benefit of having anemometer and air velocity? It was very interesting study which we published in Variable Air Velocity Within Stability Chambers, a possible cause of out of trend results. What we did was, we took 50 beakers, put ethylene hydrochloride, which are hygroscopic in nature, at 50 locations in the chamber, on different shelves, different locations, in the east shelf. And you see over here, what was the air velocity fluctuation, the influence of air velocity on moisture gained by ethylene hydrochloride. Figure 4a shows the moisture gain profiles and figure B shows a plot of rate of moisture gain versus air velocity. As the air velocity was high near the fans, there was much higher moisture gain. At the corners in the chamber, air velocity was low, dead end, sharp edges. There was very less moisture gain. Now if you are handling a blister pack with PVC which is semi-permeable and you place a lot at the site where the airflow is very low, your moisture gain will be very low. If you put a fan near the fan where there is a fresh air coming, if it is throwing inside and there will be always a fresh air coming on the product, the moisture gain will be much higher. Now this is another ignored parameter with respect to stability chambers. There is no air velocity mapping inside the air chamber, in the stability chambers. And this publication, you know what it is dated? 2000. 2003-04 that we did the study in our lab. Hanji.
with this meter we can map it, but how it can be avoided? Because without pen we cannot control the things.
I had put this paper in Pharmaceutical Technology journal, which comes from US, Advanced Star publication. They were so happy to publish it without any comments coming. Very interestingly, one company in Europe created a chamber where the air was coming without fan. They have created a laminar flow inside the chamber. All the air was coming uniformly inside the chamber and going from the other side. Getting replenished, coming again inside. So, means everywhere there was a uniform airflow. So, question is, until unless we have innovative minds, we look air velocity as a factor which can lead to differential uptake of moisture by the products, which are hygroscopic drugs and they are in semi-permeable packaging. See, sometimes what we see is that we see the problems in the market with our products. But we don't have the solution to this because we are unaware that light can increase the influence of moisture gain, your airflow can influence the moisture gain, and our stability chambers nowhere in the world are designed for air mapping or light added into the humidity and temperature. We continue facing the problems. No innovation mind we are putting in. This is what was NIPER different from other institutions, where we are using the stability chambers for a very challenging type of research. We were throwing ideas to the world. Then chamber monitoring network. You know that there are chambers, multiple sensors are there, go into the computer, server to computer, LAN is created, SMS are sent, emails are sent as alarm systems. So, you have to create this network and you have to select the features on offer. So, what I did, I took 10 vendors, 10 manufacturers, looked into what are the optional features they are offering and have prepared this list. So, now today there may be much more, it may be a 5-year-old list. Today if I prepare the list, there may be 10 more features they are available in the chambers. But if you do a good comparative studies, don't go blind. I want to buy Kesar. Why Kesar? Why not another manufacturer? And look into that also, what they have specification to offer. Now, anybody have listened to data loggers with 10-year battery? So, system with increased energy efficiency, now they are launching today, they are launching a system. Full feature communication, state of art data recording and trending functionality, automatic email alarm stop, web thin client for remote internet monitoring. We were talking at that moment because international vendors are talking about it. Today, Indian days people give you everything. They can enter into your programming over there and correct wherever possible remotely. So, PC-based system for alarm data logging, chart programmable, ramp SOC control profiles, touch screen interface, network rating system, high precision accuracy, multiple level password protected, 21 CFR Part 11 compliance. What are the laboratory essentials here? Utilities, water, stabilized power supply, UPS power supply to the control panels, which they are giving now. You have to provide that supply to them for UPS, for the controllers, stabilized power supply, water of their quality they require, dust-free and well-ventilated room with forced air circulation or exhaust fans for effective air circulation in the room or provision of air conditioning, distilled water or demineralized water supply to the humidity system to avoid scaling the humidity tank, and effective drainage system. Now, people ask, put it on 3rd floor where AHU is installed. There is no drainage system. Or put in the basement where there is no drainage system. After all, spent cold water which is coming out after the tank where they are having the elements, hot elements, they need to drain out, flush out the salt that is accumulated. They have the drainage system requirement. Availability of service team, parts and components. So always look for a vendor which has sufficient backup plans for you and minimize downtime. So question is, that is 24-hour excursion. Why not the company assures that there will be no excursion and work with you to create a system which is no excursion free, 24-hour. Service and preventive maintenance programs. Work with company and find out what are the best. Don't avoid on this. Give AMCs. They also have to survive. Buy and give AMC. Now, I'm constructing my house. These days I'm busy in that. For the last 4 years, it's a 4th year because scientist is constructing that house, not a simple person, not a contractor. So I have to make sure, because I have taught engineering drawings during my early days to students, pharmacy students. The other day, the person was putting the tile. I said, there's a 0.5 mm difference in your inclination, in the straightness. He took and placed practically a spirit level and found that 0.5 mm difference. I said, I will correct it, don't worry. That accurate mindset, if you have. Now that preventive maintenance, why not you help and work with them? Now I have lift at home, I have VRF at home. My son says, Papa, don't worry, I will pay you money for AMC. Don't ignore getting AMC. Whatever is AMC, if you finalize the deal, must have AMC for this system because they are continuous running system. At the need when it is so hot outside, when we came inside, I had a coat on myself and the temperature was high. We asked them to bring down the temperature. So at that moment, you cannot survive. I would have put away my coat otherwise. You could have done that. By the way, I asked these people, they came to Chandigarh to hobnob with me how the program is done and how much is your consultancy requirement. I was doing that. So what we did on that day? So we had lot of discussions how the program will run and there will be an error-free situation. It should not happen that something goes wrong with the program. So Mohali Patel brought all that agenda. They came spending money. 4 of them came to Chandigarh to sit with me to understand how the program will flow. Now I know they have overstepped the time they have due to late arrival of the guest. I said in technical seminar, never have this chief guest and everything. Let us start directly. Another thing which I have told them in the morning, which is not happened. We were 500 people sitting in the hall. Now it's at dinner time, they have to go and their half is almost half empty. I went to the security person of the hotel. You have to come over here. In London, when the meeting, I was attending that meeting. First, the security person, manager of that hotel came and told these are the exit routes. Verified that these exit routes are not blocked by the material they have put over there, chamber they have put over there. In India, who cares? We have 500 people in a hall. Something happens. Which are the exit routes? These are not the exit. It is written exit, 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 5 points. Somebody has to tell. Have you bothered to look into exit point when you came into the hall? This is our such a low safety standard. But then nobody came, I tell you. Despite my telling the team over there, nobody came to start with this. He has come from US, Canada, after study. This is the differential standard in India that you have to face. Don't run back. No. We will improve over here. Yeah, you are here to improve. Good. Okay, very quick I will go. Already late. I have given the time till 8:30. Qualification at the user side, installation, operation qualification details. You have performance qualification, maintenance and troubleshooting. This is what is supposed to be done. Additional qualification required mapping of temperature and humidity in empty and stack conditions, recovery of condition after door openings. I have told you already, decay test to determine how long the chamber remains within specification subsequent to power failure. That is another, power has failed, how much time it will retain the set conditions. Verification of standby systems, performance history review, equipment repair and adjustment log, monitoring system data review. Then network qualification, which you create the network. Now here we fail. It is to be done by IT people in the company. So it verifies that Ethernet or Wi-Fi communications, centralized data software and GSM global system of mobile communications, mobile alarm notification systems, transmit critical temperature, humidity alerts accurately and without delay. Network qualification elements are listed over here, which IT team has to look into. Not these people will be able to do it because network is created at your end. Then computer system validation, CSV, where the guidelines are FDA-CFR Part 11 and GMP 5, which have all these elements included over here. So you look into Google and find out what is GMP. Good automotive manufacturing practices. Automation manufacturing practices. Then requalification. This is also a regulatory requirement that is emerging. You are qualified. With what frequency the chamber has to be requalified? So once it is done, so European GMP next 15 recommends periodic review and requalification based on criticality and risk. So this is also an important thing which normally practice is used in 3 years or 5 years. That is that you requalify your whole chamber and use the standby chamber and requalify this chamber completely. So this is recommendation, this is PIC guideline which is going to be implemented from 1st October to 2026. So it has a clause over here, requalification. So you should go through this guideline to understand more about requalification process. This is going to be implemented from 1st October. Deciding on partial or full requalification. Now that is another agenda, whether you will go for full or partial. So it depends on formal risk assessment that evaluates how a change or event impacts the chamber's validated states. According to regulatory frameworks like EU, GMP, Nexar 15 and FDA guidelines, a full requalification repeats the entire IQ, OQ, PQ cycle. A partial requalification targets only the specific parameter or phases altered by a modification. Small modification that has been done will require partial, but if modification is extensive, it will be full. Now here there is a decision matrix I have given as a solution to you. Does it affect, when drafting the change control risk assessment, ask for these 4 questions to justify your strategy. Does it factor chamber geometry or uniformity, the change that has happened? If chamber geometry is there, does it alter environmental control capabilities? Was critical data integrity compromised? Has the physical environment shifted? So if you shift the chamber into a different physical environment, then you have to go for full scale. When to choose partial requalification? When there are small, routine, time-based requalification, minor component replacement, sensor calibration or replacement, adding a new set point. So all this, when you are from 2560 or 3065, you go to 4075 to use that chamber, you may need partial requalification. So when to choose full requalification? Physical requalification, major utility upgrades, critical system overhauls, software firmware upgrades, chronic quality excursions. If it is happening regularly, you have to go for full requalification. Now mapping by the vendor, which is very important, which I discussed, the example of giving you the car example and brought SAR over to the stage. So how many numbers of units required to use to map chambers? 9, 2 layer of 4 loggers each, 1 near each corner plus 1 logger at the center, or 15, 3 layers of 4 loggers, 1 near each corner plus 1 logger in the center at each level, or 4 or 5 loggers on each shelf. So where will each data logger be placed? They should be spaced uniformly, taking a grid map, and then you place evenly at all the places. Will the mapping take place with the chamber empty or for an operation qualification or full of product for a performance qualification? Now here very important, stability chamber is a breathing equipment. You have perforated shelves, you have to have perforated shelves. There may be wire perforation or a plate perforation. Second, you cannot ever pack it more than 75%. Even when you are packing your products on the shelf, you have to keep margin for air circulation. People have seen packing up to 90%, not allowing the air to circulate freely. You cannot, because you say, otherwise I have to put another chamber. So let me pack it as much as possible. Like we do in household fridges. Sometimes when I bring vegetable from the market, everything we push, not just trying to look into whether air circulation is happening or not. Okay, are you mapping air temperature inside the chamber, or will probes be needed to track interior product? Now the question is, how many of time you have tried during the mapping that you put a sensor inside an open bottle to see what is the product temperature inside, because it is in a primary pack. You are looking into the fact whether the primary pack is allowing the product, the temperature to go inside the product or not, or it is preventing that. So how many times you check mapping with a open container placed alongside? And in one very interesting phase, I went to a company, and always I used to go to the stability room. I opened the chamber, all the products were lying inverted. They were vials with powder inside, and they wanted to see that interaction of the closure with the product inside. And this was a European requirement that you check the product in inverted state. So how you handle your products and samples is very, very important to get to the accurate stability happening with the product. What parameter will you measure? Should the chamber be mapped for temperature only or temperature and relative humidity? Yes, it should be for both. How long will be the mapping study be? Typical study lengths are 24, 48 or 72 hours. Occasional facility map for 1 week, 1 month or even longer. Routinely 72 hours is enough. Maximum 1 week. But if there is some fluctuation you are expecting and it is a requalification, then you can go for a month, not for new chambers. Or if the walk-in has been installed, you can decide internally what you want to do. When will the mapping study start? Valid time. Determining your test schedule will allow you to set all data loggers to simultaneously start and stop collecting data at the same time. And we have this wireless charge sensors these days, and they are mapping. Kesar is also mapping with the wireless sensors. Now, there's a difference between mapping and continuous monitoring. This you have to keep in mind. Mapping they will come and do and go away. But there has to be a program and sensors placed by the manufacturer for continuous monitoring. Now, determining variability in set conditions, stability test storage equipment for month and year. When you are continuously monitoring temperature and humidity, now MKT is one parameter which allows you to monitor small variations, natural variations with the sensors. Because sensors also are placed differently, there will be small difference in the different location, and you take an average and try to give that this is a fluctuation. That is defined through MKT, in what is happening in daytime. We have a chamber, like we people are also facing morning time, evening time, night time, temperature fluctuation. So everywhere we calculate MKT to define what was the temperature in a day, what was over a month, and what is over a year or 30 years. The data which is collected, weather data by weather department, they come out with an atlas of 30 years. After 30 years, they became an atlas. I used to have in my department that 30-year data, 1930 to 1960, 60 to 90, like this. That provides MKT value. Over the 30 years, how much of the variation in temperatures has happened? Because now we say global warming. It is truly global warming not faced by Earth ever. If we have historical data, we can say, no, it is not happening. It's a routine happening after every 40, 50 years. There is a cycling. Sun cycle is there that it happens 40, 50 years. So there is nothing novelty. Same MKT is used for that. What Waterman has proposed, this is one mean kinetic relative humidity, a new concept for assessing the impact of variable relative humidity on pharmaceuticals. He also should be given Nobel Prize. He has proposed MKRH also. Unfortunately, the regulatory bodies have insisted MKT, which is defined in the new guideline also, MKT. In the ICH 2025 draft, they have given the definitions of MKT. But not working on MKRH. The equation has already been developed, similar to MKT. Now, if you people in India, in pharma industry, use this equation to find MKRH along with MKT to show to the regulatory, they will be surprised. You are so advanced that we are still at MKT, you are working on MKRH. But who will read that there was a paper published in 2007? How much? It was in 2012 in Pharmaceutical Technology that it was published, this paper. So, now influence of recording frequency. Now this is another question. I don't know how you handle this, recording frequency. Now if you have sample interval in which you record every 10 seconds, the inset conditions, temperature of humidity. One channel you need 4 days will override your storage. 2 channels in 2 days it will override. 1 minute, 24.3 days, 12.1 days. 5 minutes, 4 months, 2 months. 15 minutes, 1 year, 6 months. 1 hour, 4 years, 2 years. So you have to look into what is the storage capacity of your memory banks in the chamber. Accordingly, you have to set. See, in a 5-year long study, if vaccine is being developed in a hurry, I will have 1 second or 5 seconds. Only, let us say, short term storage is needed. When you have a span of 3 years to 5 years, why not have 1 hour data, 24 study points are good enough. Calculate MKT, weekly or monthly, is good enough. So this has to be a rational decision based on the memory you have. You don't allow memory to override because then it will stop storing. Very important point. Then stability, sample charging, storage location, withdrawal, inventory control, etc. This is another very, very important point in stability handling. So determinant factor, number of samples to be charged to stability chambers. There are multiple factors. But one, I give you the Asian guideline. They have calculated total 13 boxes or 10 blisters, which are talking 1300 tablets for all long-term study, intermediate studies and accelerated study. So now these 13 boxes have to go inside the chamber. You have to open them. You have to withdraw the samples. You have to define the location. You have to define the lot, how many lots you are manufacturing of each batch, how it will go, create capacities accordingly. So charging of samples, market ready container enclosures, including secondary packs, duly labeled with the date of storage, withdrawal and condition, prevent commingling with other products or labels, prevent commingling with other lots, pre-charge approval, charge verification. So all this has to be done, which is a requirement, regulatory requirement, and people from inspector will like to see your data. Sample pull issues, well controlled, consistent, SOP driven, defined month or year. Now I went to Turkey on this consultancy trip to give a 2-day lecture in one of the companies, one of the second largest company in Turkey. The day I was giving a lecture, that was the date of withdrawal of the samples. Date of withdrawal was the date when I was giving a lecture and everybody was in a lecture. There was nobody to pick up the samples from the chamber. I said, why this is happening? They said, because the training program put a deviation over there. Now, you have to set a date. You have to make sure Hammonds may turn, but you are withdrawing the until there is a disaster happening. Not SARANJIT is not a training, is not a disaster. It's not defined by regulatory as a disaster. So that way, cleaning is easy. Once you have all the samples going in 1 month, specific date in 1 month, you have minimum chamber openings. There's minimum variation that is happening, deviations that are happening, and minimum justification in audit trail. So these are important points when you are handling the chambers. Manual or electronic, both are acceptable. Regular pick list, daily, weekly, monthly, that you have to decide. Prevent commingling of same or different storage conditions. Labeling samples after the pull. Individual individual sample prevent mix up downstream in the sample handling areas and lab. Ziploc bags, a set of segregated sample by lot and storage condition. Then they are transferred to the laboratory. Sample pull out and testing windows. I told you about windows. Normally 7 days windows are the industry standard in international companies. 7-day windows are between manufacturing and charging the. Now this is also very interesting. I listened and understood what the word charging that is used in industry. As an academician was not knowing, we say put the sample in the chamber. There's no, chamber has to be charged. That was the terminology I learned from somebody in the interaction of this kind. So question is, it's a 3-year-old study. In the first year, you have to set a 7-day window of sample charging after manufacture and sample analysis after pull out. If this is to normal variation, but it is important. If you are starting late, let us say, you have manufactured the product and there is a gap of 1 week, doesn't matter because the product is lying at the room temperature 25-60. You have to go to harsher condition, it is lying at a normal temperature. No question will be asked. Second is when you pull out from the chamber and for analysis, if there is a delay, 1 week, you have kept at a normal temperature, fine. When you are plotting, I always put a question, why you are worried of windows? Gap between the pull out and the analysis. Because the day you are doing the analysis, the value is, you are obtaining the value of SA over there or impurities over there. When you are making a 5-point plot, why don't you put the exact date? 3 months, 7 days. 6 months, 7 days. You can plot easily. Instead of putting as 6 months, put 6 months, 7 days. Very easy. So where you need window system, you don't need window system. When you are analyzing, let the chamber continue. Don't withdraw until unless you have a set date. If you have not set a date, your system is that you can withdraw as per requirement whenever the person is available. Suppose person is on leave, you are not withdrawing, he knows the job. Yes, come, okay, let the sample be there in the sample. You have gone after 1 week for a particular lot, you have brought it out and tested. Plot 3 months, 7 days, 10 days. What is the harm over there? You are not doing anything scientifically wrong. So this type of understanding is very, very important. So sample puts and testing windows, that is just inventory control from receipt and final disposition, that should be there, receiving records, transaction history, physical inventory. Then coming to risk management. Now every guideline is focusing on risk management. So 2025 guideline also is this. The systematic process of identifying, assessing and mitigating threats. Equipment and environmental deviations can ruin years of testing data. Any deviation that happen at any stage of study, long-term study, will ruin your data. So mitigation through strict equipment qualification, redundant temperature humidity controls and continuous 24/7 automated alarm systems. You can avoid this situation. Capacity constraint, running out of shortest space during unexpected product launches or new studies. You have planned everything, but suddenly requirement came that you have to have new launch, set date, you don't have the capacity. How you handle this situation? This is a disaster situation. Mitigation, regular capacity forecasting by monitoring historical trends and setting alerts when chamber reach 80 to 90% capacity prevents overcrowding. Unauthorized access, mischievous action with the disgruntled employees. You are not given promotion to a person. I said, your samples are lying for 5 years, I will go and disturb that. You have all access controls. He was given, he has that, but he is disgruntled employee. Want to create mess out of it. We can do that. It's happening. So, mitigation control access implying access codes, swipe cards, keyed locks, security guards, etc. Additional reliance on electronic supplies, security system like CCTV, motion detectors. So why is going at odd times to do the disturbance? Daytime he will not do. He knows that he will be captured. Have CCTV cameras in the whole area. So all preventive steps have to be taken. Power outages and disasters. Electrical failures, fires, flooding and other hazardous weather conditions like hurricanes, tornadoes, earthquakes, snow or ice storms. This is not prevalent in India, but it's prevalent in coastal areas in India or US it is happening routinely. So mitigation requires emergency backup generators, uninterruptible power supplies and pre-validated contingency storage of an off-site partner location. That is one solution to this. Then regulatory non-compliance, lacking document backup plans or failing to follow GMP standards can lead to audit failures and rejected during drug application or rejected drug application. Maintaining strict standard operating procedure ensures complete traceability and data integrity so that regulatory non-acceptance is not there. So question over here is, okay. One company, Japanese company, Nagano Science, offers complete risk management for you people. I don't know anyone is doing in India that I take over of your risk management. I will plan out everything for you and set up the whole facility, whether it is an off-site facility or whatever it is. So this kind of concept is very importantly coming now, and regulators are looking forward to this type of facilities available. So friends, finally, in-house manning of stability storage facilities. Unless you have stability managers, you have maintenance technicians, you have stability technicians to monitor stability chamber, document deviations and assist in execution of contingency plans, and quality assurance team attached to the stability team. You cannot run the facility. So you have to have a very strong stability manning setup in your company. So before closing, it came to my mind that I'm going to audience of such large number. What is future in stability testing storage facilities? I was very happy to go to the website of this company and found that they have a excellent blog write-up created by their people. Kesar, the future of stability testing, tends to watch out for. Now what they have put over here in this, trend 1, IoT driven stability chambers aligning with the smart factory initiatives. Second, green technology for sustainable testing, the shift toward energy efficient equipment which they have launched today, congratulations to them. Trend 3, AI powered analysis and automation. which is enhancing accuracy, because AI will give you the whole trend analysis of what is going with the chamber. If you ask AI, this is my data, and what is the deviations, extent of deviations or fluctuations, whether it's a deviation or fluctuation, they will give every answer. Stricter regulatory standards worldwide, even readiness to global regulatory changes, tailored and modular chamber designs, the need for custom solutions. So these are the trends which they have listed. It means this company is thinking ahead and advance, and maybe their now innovation and R&D teams are going to come out with the chambers with IoT, AI, and this will be very interesting to look forward to from Kesar. That is all for the day, friends. Thank you all so much. Jai Hind. So, any questions you have, pendency is there related to the topic, not general stability testing. Today I am not talking of stability program as a whole, because this is the first seminar on the topic. I don't want to deviate. So if anybody has any question in mind, please raise hand so that you get stand up. If anybody has a question, please. I have tried to cover, yes please, as much as possible from my academic style.
Yeah, thank you, sir.
Thank
for
you.
Very insightful session. So how we can address to the regulatory auditors if any disaster happening in the plant? So how we address the ongoing stability samples?
See that some solutions I have provided that off-site you create a situation that you know that this is a period when disasters happen normally, like hurricanes. US people know that this is a period most hurricanes happen. One odd situation and odd happening in the period throughout the year is different. So the solution I have provided over in the slide, that you have to predict based on the trending and whether I can put my samples, one set of samples off-site to have a secured material available for analysis. Regulatory people, and then I have told you separate UPS, separate electricity, so the electricity failure is there. Till it comes out, UPS capacities, how many are there? We have to look into the holistic plan, what action plan we can do in such situations. There are multiple possibilities and regulatory will be convinced. They know कि यार इसका तो सारा data loss हो गया, ठीक है ना? But now, suppose you are making batches, you have 10-year history of making the batches. 1-3 batch which was in the stability chamber, they will get convinced, sir, हमारा 10 साल का record ये कह रहा है same batches का, it cannot be very different. So this is the way you have to explain to regulatory. There is no other way, and they will be sympathetic in that situation.
Because recently heavy rainfall happened in Ahmedabad, and we are facing such challenges. We search.
You have your chambers in the basement?
No, in first floor.
First floor, what was the challenge?
So flood was there. So entire campus affected with the flood.
Okay.
And around 10 days we shut down the power.
Acha, power shutdown was
Yeah.
there.
So there is no power
Hey,
backup.
power shutdown was from the back, from the torrent, or it was?
No. Entire plant disconnect from the power.
So what how do you handle the situation? Let us say.
So we decided.
Do you feel what I have suggested could have been implemented?
We are in discussion with the rented chambers for meantime. Till the time we will.
Okay.
Recover
Now,
the
say
story.
if I am again saying, if there was a flood, there was a 10 days no temperature. So you have a set of data available.
Yes.
Last sample analysis.
Yes.
Okay. 10-day gap has come. Fine. Now the break is there. Either you continue with the study after 10 days and plot that delayed point. Okay. No electricity means the temperatures have come down, humidity has come down. There is not that serious adverse effect on the product. You continue the study after 10 days, try to plot reducing that period, minusing of 10 days. What are the best options? Do brainstorming. What are the best possibility to convince? Historical data of the same products, that is one. Try to plot with a gap of 10 days whether you get the same slope with the previous batches, yes or no. So try all the means possible to convince the regulator.
What about 2 to 8 storage?
What?
What about the refrigerated product 2 to 8 storage?
Yeah, that is a challenge. You have no answer. We have no answer to that. If you have biologicals in a refrigerator and the temperature goes, you have to only rely on historical data to convince the regulator that this study failure has happened. We are now this batch we are releasing based on historical data on this product. That's the only way. No other way. You can suggest any other way?
There are like two situations. One is the worst case scenario. So there is excursions which has happened.
Which has happened
which
beyond
is like
24
going
hours.
above. Which is
Going
going above. Then it's a worst case scenario. By chance if it is passing, in a worst case scenario it pass. So obviously it is passing in a normal conditions. If it is lower, just extension of the durations, and that will serve the purpose.
What they are saying is refrigerated
2 to 8 degree.
when the temperature 2 to 8 degree becomes a normal temperature humidity condition. So in that case, test the products. Test the products.
Correct.
Whether there have been a change in trend value, discard that result. If there is no change in trend value, the product still sustains as it. Like I tell you, my wife is using Manjaro. Okay? So it says Manjaro has to be kept in a refrigerator initially. Beyond that, the injection is to be stored at room temperature. Okay? So for long-term storage, but in use, when you are using it, it has to be kept at room temperature. Now it means they have tested the product in refrigerator also and in use condition that it should be at the normal temperature, not extending beyond 30 degrees centigrade. So whether your product was of that kind, that which can withstand normal temperature of short duration, additional data if it is available, excellent. If it's not available, you have to discard the study. So for you, 2 to 8 degrees centigrade, the accelerated condition is 2560 or 3065. If you have that data available of previous batches, and then you do testing after 10 days at 2560, bring it down to 2 to 8 degree, take a sample and subject to testing whenever the time is there, time allocation when you have to withdraw the sample. If it is showing lot of out of trend data, discard it. If it is showing in the line, because it was stable at 2560, like 2 to 8 degree it was stable, it is stable also for short time period at 2 to 8, 25 degree, you can continue. You just should have data to convince the auditor. Without data, no. They will reject your study. And that should be well thought of, based on the historical. Stress testing data again.
So in a regular testing of 2 to 8, as you said, in that 7 days testing delay. So in that time also we should keep in the refrigerator, or we should pull out if it is storage is 2 to 8.
I could not understand.
If the long-term storage is 2 to 8 degrees centigrade, and the testing delay may be 4 to 5 days, 7 days.
What delay?
Yeah.
In biologicals, don't go for that.
Okay.
Because the short term, it is a total study duration is short term. It's not long term. Don't go for windows.
Okay.
Avoid windows. Or reduce the windows of 1 to 2 days delay maximum.
Withdraw only the time of testing.
Yes.
Yes.
Because you have to squeeze the time of testing, and then you have to pull out and keep in the fridge again.
Right.
Or you don't withdraw.
Right.
That's the best. Don't avoid windows in a short term period.
One more question about your presentation, sir. It was very interesting that in presence of light, the water absorption is increasing. So what could be the mechanism behind it? What do you think?
Yeah, this happened when we set out first paper in International Journal of Pharmaceutics. It was accepted. Second paper, part 2, that we put on the excipients and drugs, we sent to the same journal for continuity purpose, or for readership. It is easy. They said, tell us how it happens. why it happens. Now, my answer to this is that light is an energy. The molecule, ethyl molecule is very symmetrical molecule. Okay. The bonds are at one stretching point, uniform. When the light extra energy we provide, it starts vibrating. And with that vibration, it gains more moisture. Maximum rate is increased, but total moisture capacity is same. I went to physics department at Punjab University to find out if anybody can tell me how I can calculate the vibration model in molecules. They said we don't have such a device yet. So I could not publish in International Journal Pharmaceutics second paper. So I had to go for magazine, Pharmaceutical Technology.
Yeah, by maybe, as you said, by any mechanism there may be a surface polarity increase which can attract the water and
some
hydrogen
vibration
bonding,
increase that it attracts more moisture.
Yes.
Energy, light is an energy.
Correct. Thank you, sir. Anyone?
I am the manufacturer of medical devices.
Oh, good, excellent.
Yes.
Congratulations.
So in this we complete the stability in 25 degree. As per EU MDR now requirement, upper limit and lower limit we can mention on the label. So upper limit we have done the stability 25. So how can we write out the lower limit of the temperature?
Okay, interesting. Now the question is that what are the guidelines for these devices which you follow?
ICH guideline.
ICH guideline will not apply directly to them.
Because
I tell you, you are talking about limits in the sense that if it is a normal temperature is 25 degree high temperature, your low temperature.
How to mention, because our product is the room temperature. We claim the 25 degree maximum temperature you claim.
No, I have to understand, Sej.
Yes.
You are saying that your labeled storage condition is 25 degree.
Yes.
You are testing the product 25 degree. Are you going for accelerated?
Yes. Accelerator we have carried out in 40 degree.
40 degree. Now the question is why you want the lower one?
Because UMDR now requirement is the symbol mentioned. You have compulsory mention the lower limit and upper limit for storage condition.
Then see here the
Range.
nature of device, whether it's a metallic, plastic,
No,
it is a stent with drug loading.
No,
What kind of device it is?
This is a drug, injected.
Injectable
drug.
Injectable drug.
Orthopedic injections.
Huh?
Orthopedic injections. Sodium hyaluronate injection.
Pre-filled injection.
Pre-filled injection.
Pre-filled.
PFS.
So now the question comes over here is, if it is a pre-filled injection, what are the storage conditions for the drug that you have pre-filled in a syringe?
25 degree.
25 degree.
Yes.
So here, what is meaning the lower? Are you recommending the store in a cool place at not?
For sale in Europe, we must put the symbol for upper limit and lower limit as per you.
नहीं वो अगर लिख रहे हैं तो तो इसका
already we have write the do not freeze sun, keep out of the sun.
नहीं आप आपको वो फिर frozen
If I
temperature
put then 15 degree at 2 degree, then auditor required the stability to be put down in 2 degree.
मैं वही कह रहा हूँ मेरा पॉइंट ये है कि हमारे पास एनवायरनमेंट में एक हाउस होल्ड में या एक फार्मेसी क्लिनिक में या क्लिनिक में फार्मेसी में या क्लिनिक में जहाँ पे प्रोडक्ट सेल होना है या यूज़ होना है कौन से स्टोरेज कंडीशंस अवेलेबल हैं वी हैव ए रेफ्रिजरेटर ठीक है वी डोंट हैव 15 डिग्री एवरीवेयर वी डोंट 25 भी नहीं इंडिया में तो 35 है यूरोप का 25 चलेगा तो वो कह रहे हैं कि आपने एक्सेलरेटेड टेस्टिंग की कि इसका टेम्परेचर 40 डिग्री पे ख़राब हो जाता है प्रीफिल्ड सिरिंज है प्लास्टिक है लीचिंग होगी जो भी होगा एक्सट्रैक्टेबल होंगे लीचिंग होगी सो यू अवॉइड गोइंग टू हाई टेम्परेचर लोअर टेम्परेचर आपने फ्रीजिंग करनी है नहीं रखना क्योंकि फ्रिज है आपके पास सो 2 to 8 डिग्री पे आप रख के देखो उसका कारण क्या होता है कि यू हैव ए प्रीफिल्ड सिरिंज will your material inside which is in a liquid form precipitate out crystallize out become hazy इसलिए रखना नहीं है उसको. So question is, you have to test in the refrigerated condition, you have to test at the 25 degree chamber, you have to test at 40 degree chamber, accelerated condition. तो आपने उस पर जब टेस्ट किया लोअर एंड हाई, if there is no change at 2 to 8 degree, rather you find increased stability of your drug, prefilled syringe, drug in the prefilled syringe at 2 to 8 degree, then you say product without use, जैसे मैं बता रहा हूँ इसमें लिखा है, आप मंजोरो का इंजेक्शन का वो ले लो ना एग्जाम्पल, वो भी प्रीफिल्ड सिरिंज है, उसमें लिखा है नॉर्मली दैट व्हेन इट इज़ मैन्युफैक्चर्ड पोस्ट जो सारा हैंडलिंग है इट शुड बी डन एट द फ्रोजन कंडीशन 2 to 8 degree, not frozen but 2 to 8 degree. But when in use for 3 months or 1 month use, 1 month के लिए सिरिंज होता है, keep it at normal temperature because it is stable at less than 30 degree when it is in use. क्योंकि फ्रिज में निकालोगे रखोगे बाहर, फ्रिज में अगर प्रेसिपिटेशन होगे तो क्या करोगे, तो वो क्योंकि इन यूज़ है, खुल चुका है उसका सारा कुछ कंटेनर, उसमें क्लोयर में पियर्सिंग कर दी आपने, तो ये आपको based on the nature of your product and doing the study at lower temperature of 2 to 8 degree or even आपको लगता है कि 2 to 8 degree